Hi, everybody. Good morning, and thank you for staying with us again for the second day. I hope you all had a wonderful day yesterday and you were able to get some rest. And we are ready to start for our day two proceedings. The sessions today, and we're actually going to start with one of the sessions that everybody in our leadership team has been really very interested in, that is neonatal neurology neuroprotection. We have a lot of things to discuss specifically as they pertain to challenges in therapeutic area development. Where is the field as it pertains to preclinical work for neonatal neuroprotection? What is the current status of the clinical work in this area? And this session is going to be moderated by our colleague from FDA, Office of Pediatric Therapeutics, Anne Massaro. And the presentation is going to start in just a few minutes. Anne, are you ready? Awesome. I'm not going to take too much of your time, but welcome, Anne.
Good morning, and thank you, Kamaljeet, and to the whole program committee for allowing us to talk about this topic and issue today. And it's wonderful to be here and meet many of you in person for the first time. Being a new face to Inc., only seen and heard from many of you by Zoom, so it's really a pleasure to be here. So as Kamaljeet mentioned, this morning's session is going to be on neonatal neuroprotection, and these are my standard disclosures. So I'm really going to briefly set the stage for our discussion today. So I'll start with reviewing some of the various etiologies of brain injury in the term and preterm newborn, but really more discuss kind of the paradigm with which we think about injury to the neonatal brain. Then we'll talk a little bit just to describe the current landscape of neonatal neuroprotection in the NICU, because of course this is the context by which we're going to be thinking about any of our future trials. And then we'll discuss or touch upon, introduce some of the many challenges we have for development in this area. So any talk on neonatal brain injury really starts with this figure, or a version of this figure, now published almost 20 years ago by Donna Ferrero, one of our pillars of neonatal neurology, to just really describe somewhat simplistically, but really aptly, the process of brain injury or the propagation of brain injury in the neonate. So we start with an acute insult, whatever that may be, and then you have an acute period where many of those physiologic processes, oxidative stress, excitotoxicity, many other cascades are triggered. And then there's a subacute phase where we know that inflammation plays a really important role. And all of this culminates in cell death by different mechanisms, apoptosis, necrosis, new cell death mechanisms that we're learning about every day. And then added complexity of the brain's capacity for repair. So when we broaden that conceptual thinking to think about developmental brain injury, where we're really talking about injury to the immature brain. So not just the neonate, as we have that narrow definition, birth to the first month of life, but thinking about brain development along that continuum that starts during fetal life to term and beyond. So the resulting neurologic dysfunction that we see, again, is very complex because it depends on a lot of different factors: the type of insult, the severity of that insult, how long it lasts, and of course, as we're talking about developmental brain injury, the specific developmental stage during which that injury occurs and or continues to occur, and the capacity, as I mentioned, for plasticity and repair. And again, as I mentioned, we know that the brain is developing, and we're here as Inc. and neonatologists thinking about the neonate, but remembering also that this process continues through infancy and early childhood and adolescence. So this very complex continuum is what we need to think about as we think about both the insult and the interventions that we might provide. With regards to different etiologies of brain injury, of course, there's a whole laundry list. This is not, all inclusive, but some of these conditions that can occur specifically in the neonate versus conditions that have adult correlates, but obviously different manifestations in the neonate, whether it's hypoxic ischemic insults, infection, inflammation, strokes, hemorrhagic injuries, metabolic disturbances, and then some of our injuries, of course, that are that are more specific to or common in the preterm infant are IVH and PVL. This figure from a recent review article on preterm brain injury is just to remind us that we don't always select one of these conditions or etiologies. And remember that in the months that our preterm babies are with us in the NICU, they can be exposed to many different insults, from infection, inflammation, ischemic insults that all culminate into the brain injuries that we see in the preterm infant, and in any of our infants really that spend time with us in the NICU. So to provide that historical context or overview of where we've been and where we've come to, I think one of the forefront acknowledgements is to say that we have no approved pharmacologic agents with an indication for neonatal neuroprotection in the US. Clinical care largely remains supportive, with the exception of therapeutic hypothermia, of course, which I'll mention. And most of our intervention trials in this space have really focused more on clinical care bundles. As neonatologists, we know that we all have a lot of variability in what we do and how we care for these babies. So a lot of trials have focused on, how do we do that golden hour for the preterm infant? Is head position a risk factor? Then can we do some changes to our practices that may be helpful for these high-risk babies? Some of our pharmacologic trials, I hate to use that word, failed, but really have, not met the efficacy endpoints for which they were being studied. So IVH, our devastating injury in the preterm infant. We've studied several different therapeutic options there, both on the prevention side and on the treatment side, that have not translated into viable therapeutic options. And of course, more recently, trials in the HIE and preemie neuroprotection space as well, with Xenon and EPO, that our speakers, I think, will touch a little bit more in detail on today. So I mentioned therapeutic hypothermia before. I'll say this is our one major success in the last several decades in this space. And I think most are familiar with, again, the two pivotal trials that were reported nearly 20 years ago now for selective head cooling as well as whole body cooling, and really have become the standard of care. And I think it was talked a little bit about yesterday when we think about, natural history-based or real-world data-based studies, and thinking about control groups and what are the appropriate control groups for these studies, and knowing what the current standard of care is. Obviously, I think it was mentioned yesterday, having a normothermic, control group won't make sense when we're talking about how care is provided today for these babies in the NICU. And of course, the advent of hypothermia really has sparked a lot of development on the neuromonitoring side of things. We have, a lot of more devices in the NICU. We like our devices for brain monitoring with continuous video EEG, cerebral neurs, neuroimaging, and of course, all our cooling devices that are now on our NICUs. So again, thinking about this context when we think about future trials is going to be really important. And also to think about some of these tools that we have as potential pharmacodynamic and potentially surrogate outcome markers, which I think some of our speakers will talk a little bit about as well. So where do we go from here? So again, going back to our paradigm that I started with, realizing that this is a continuum of injury. So there's many places during which we can intervene. Of course, the Holy Grail would be if we could prevent injury. One of my early mentors described treating HIE instead of trying to prevent it is like trying to catch a burning plane falling out of the sky. I think that there is some more that we can do than that in there, but prevention obviously is a big focus. Obviously intervening, because we can't always predict when a baby is going to suffer brain injury. So we still need our post-injury interventions. And of course, those that present later to us that we don't capture right away, we need to think about reparative and rehabilitative therapies. So I'll just point to this recent review article, and Malloy wrote on behalf of the Newborn Brain Society, for kind of the future of neuroprotection. Again, just to illustrate in a different way that paradigm, that there's a continuum and many places we can intervene. And the more we think about this, the more we realize it's probably not going to be a single magic bullet. And it's going to be a combination of things that you cocktail that we throw in there, depending on where we're catching the baby in injury and where we can continue those therapies to change the trajectory of their course. There are many things in the pipeline. All of these, again, not an exhaustive list, but pharmacologic therapies, devices, biologics that you can find searching clinical trials registry for ongoing studies for neuroprotection. So there's a lot of activity, but there's a lot of challenges. So one area of challenges amongst all of these things that I mentioned are in the pipeline and other novel therapies. How do we select the most promising agents to move forward and invest in our pivotal trials? So this will obviously require good animal models for proof of concept, and then accelerating early phase clinical trials so that we can know early which therapies are looking promising, and just as important, which ones should not move forward. And as we've been discussing over the last few days, in general, neonatal trials are challenging. I don't have to kind of say a lot to this audience. That's why we're all here. And a lot of the factors are not specific to this disease area. So, a lot of these conditions are rare, requiring multi-center studies. It's a vulnerable population, as all of our neonates in the NICU are a vulnerable population. But some of the specific challenges in this space are, patient selection. I mentioned various etiologies, knowing which babies are injured, knowing the types of injury they have. All of that is key when we're designing trials and selecting patients for intervention. We talk about that, our hypothermia trials, as a huge success, and that we had this homogeneous population, but it's not a homogeneous population at all. We know that. And that's probably really responsible for that significant proportion of patients that didn't respond to therapy. So we need to be able to parse that out better than we can now. And then our endpoints. So obviously with most of our neonatal conditions, this is true, but especially with developmental brain injury, we're measuring the impact of what we're doing in the neonatal period with an endpoint that's going to be much farther down the road. So these studies are inherently challenging, difficult from a feasibility perspective, and of course, very expensive. So we want to be smart. So our session today, hopefully with that background, I'll introduce our wonderful speakers that will shed light and share some of their perspectives on these issues. And we'll start with Dr. Nikki Robertson from University College of London, who will talk with us about the early phase development, preclinical models, and then Dr. Sunny Jewell, who I think when we first invited her, she was division chief, but now she's moved on to being big research pillar at the University of Washington and a big contributor to our field, and followed by our panel discussion from multi-stakeholders and to discuss different perspectives. So I'll invite Nikki up next.
Well, good morning. It's a great honor and pleasure to be here at this excellent Inc. meeting. And I'm very happy to share with you some of the work I've done over the last 20 years. I'm a neonatologist and a scientist, and I've led the preclinical piglet lab in London for the last 20 years and set up another second lab in Edinburgh, my alma mater, in 2020. So I'm going to shift between preclinical and clinical in a way. I have no disclosures. And my talk is in two parts, and I have a suggestion at the end. The first part is really understanding the disease. And I'm going to take you back to the 1980s just to get some of the foundation concepts about birth asphyxia or neonatal encephalopathy. And I'm going to use an example of using a imaging biomarker in a failed clinical trial. I then want to share with you some of my work over the last 10 years trying to translate a therapy into from bench to bedside. So preclinical models of term brain injury have been ongoing for the last 200 years or more. We knew from Le Galois all those years ago that males were more vulnerable than females, and that temperature, high temperature during hypoxia ischemia was damaging. Taking us forward to the 1950s to 1970s, the primate models were very useful in understanding that the pattern of brain injury depends on the type of insult. And what was so eloquently described that all those years ago was the acute total asphyxia and the chronic partial asphyxia. We move on then to the 1980s, when the Rice-Vanucci model was developed as a relatively cheap platform in which to assess neuroprotective therapies. But it's important to say that there are some fundamental differences with some of these models compared to the complexity of the human brain in terms of when the brain growth spurt occurs, and also the complexity of grey and white matter and cortical folding. We move on then to the studies in the 1990s in the large animal models, the sheep and the piglet, that looked at that gave so much confidence and the concept that cooling might be safe and protective. And this was a real catalyst towards the cooling trials in the early 2000s and the adoption of cooling as a therapy for moderate to severe neonatal encephalopathy. So one of the key points I wanted to talk about was some work that my friend and previous colleague and mentor, Dennis Azzopardi, did way back in the 1980s. He took one particular baby with birth asphyxia down to the magnet, an MR spectrometer, looking at ATP peaks within the brain in a living baby. He took this baby down to be studied at 8 hours, 31 hours, 36 hours, and 55 hours. And you can see the arrows at the bottom as to when this baby was studied, and you can see what he was seeing before his eyes was the evolution of what we now know is secondary energy failure. On the right-hand side, you can see then we took what we knew in the baby and tried to understand it in the context of a preclinical study, preclinical model, the piglet. And we then were able to define the primary energy failure during the hypoxic ischemic insult and the recovery during resuscitation in the latent phase, that's on the top panel on the right, and then the evolution down to secondary energy failure if there was no intervention during that latent phase. The key point of this is that if you give a therapy in the secondary phase, when the energy failure has already started or become established, you will not be protective. What also was seen from these studies was that the neonatal encephalopathy is evolving over the first 3 to 5 days. There are different pathogenic mechanisms going on leading to the brain injury during this time, and also the evolving change in the EEG. So in 1995, an experiment was done in the preclinical piglet model whereby cooling was started during that latent phase, and that's on the top panel there. And you can see that the trajectory of the energy failure was changed in the blue, and you can see the blue spectrum with healthy ATP peaks if cooling was started before the secondary energy failure. What was key about this finding was that the energy failure in these babies in the first week related to long-term outcomes at 1 and 4 years. And so this was the first surrogate outcome marker that we were able to predict outcome subsequently. But as phosphorus spectroscopy is not available on all MRI systems, people then focused on proton spectroscopy. And we've spent a lot of time validating the biomarker lactate to N-acetyl aspartate. You can see in the green box, there's a normal spectrum, and in the red box is a spectrum from a baby with severe injury with a high lactate and a low N-acetyl aspartate. We saw that if we sampled the area in the deep gray matter, the basal ganglia and thalamus, we were able to predict outcome from this 14-day scan at 2 years with a very high sensitivity and specificity for motor cognition and language outcome. And so we now know that lactate to NEA is actually our best biomarker of outcome. And you can see that if you start cooling early in that phase, during the latent phase, you can change the trajectory of energy failure based on lactate to NEA. Of note, it's important that it's acquired at a long echo time, so you don't have the interference of lipids at short echo times. And of note, it is pragmatic. You can acquire it over 6 to 8 minutes at the end of your structural scan. In a multi-center study, Dr. Thiel and colleagues looked at the predictive value of lactate to NEA, and again, it was seen to be very sensitive and specific in predicting 2-year neurodevelopmental outcome, and more so than conventional brain imaging. And so what we have here is a bridging biomarker. We have lactate to NEA, which predicts early on what a 2-year outcome is going to be. And we spent some time validating it in the piglet to show that it relates very closely to cell death, inflammation, and caspase damage. So we took this bridging biomarker and together with Dennis Azzopardi, started to do a study to try and see whether an imaging biomarker would help speed up clinical translation. And this was because it was realized that although cooling is one of the most significant advances in neonatal care, cooling is not the silver bullet. And so we returned to the bench after going to the bedside with cooling to try and understand what therapies could be used to augment cooling to improve protection in babies with neonatal encephalopathy. Cooling does reduce the incidence of cerebral palsy and the severity of cerebral palsy. However, despite cooling, around 20% of babies still have CP, and at school age, around 60% have cognitive, behavioral, and language problems. And this is seen in brain imaging as well. So this is the modern day evolving injury in neonatal encephalopathy picture, suggesting that it is absolutely key that if you want to target the early high cascade of injury, you need to use therapies that start within the first 6 hours or so to prevent the free radicals, then leading to that cascade of mitochondrial failure, cell death, and inflammation. And xenon and melatonin are the two I'm going to discuss today. Of course, there are other therapies that we need to keep an eye on in terms of therapies to help with regeneration and repair. And of course, stem cells are a key part of the regeneration and repair subsequent to the early secondary energy failure. And that's something for another day. This is what I'm working on in Edinburgh. So the TOBY xenon trial, this is an example of a bench to bedside trial using an imaging biomarker. We looked in 2011 at xenon in the lab, looking at secondary energy failure. In the bottom left-hand corner, you can see that using xenon and cooling, we completely did away with any secondary energy failure, and it was significant compared to no treatment at all. Importantly, it was not significantly different to single therapy of xenon or cooling by itself. Nevertheless, funding was obtained for the TOBY xenon study, a phase 2 imaging biomarker study with a power calculation based on lactate to NAA of 138 infants. We know the primary outcome was reduction in the lactate to NAA. We also looked at the diffusion MRI. We recruited babies from January 2014 to September 2016, 92 infants, and there was no difference in the lactate to NAA in the basal ganglia and thalamus. This was a highly difficult project to do. The feasibility of giving xenon was questionable. We needed a specific ventilator that scavenges xenon. It's so expensive. In the preclinical studies, we used 50% xenon. In the clinical trial, for practical reasons I can go into later, we used 30%. And babies couldn't start xenon before they were coming to the specialist hospital, and the mean age of starting xenon was 10 hours. This is too late, especially for its mode of action as working on the high cascade area of excitotoxicity. However, all was not completely lost. You can see that it was 5 years between the clinical publication of the preclinical study and then the publication of the biomarker clinical trial. But at least the negative phase 2 biomarker trial prevented the move to a phase 3 study. And these babies were all followed up and had 2-year Bailey outcomes. And we were able to validate the lactate to NAA with 2-year outcome, showing that it's a very tight predictor of 2-year outcome. So moving back to the UCL piglet model, which we've been working on for the last 20 years, we continue to use lactate to NAA as one of our primary outcome measures on which we do all our power calculations. In addition, we use EEG recovery, AEEG recovery, and of course, this is standard of care for babies who are undergoing neurocritical care. Our piglets are 2 kilos around about, and so we can use all the same equipment that we use for neonatal care in our piglet lab. We have continuous EEG monitoring throughout. We have cooling, and we take the baby piglet to the 3T MR magnet to acquire the same imaging as we do in babies. The studies last for 72 hours following a transient hypoxic ischemic insult, and we are able to look at around 10 to 12 different brain regions for protection. This is our piglet neurocritical care setup, and just showing you taking the MR compatible incubator up to the 3T magnet for the imaging biomarker. We're very interested in melatonin. It's really repurposed therapy. However, we know that brain protection only occurs with pharmacological levels 10,000 higher than physiological levels. It's very potent at scavenging free radicals. It's anti-apoptotic, it's anti-inflammatory, also works through receptors. However, because of its poor solubility, which is actually one of the reasons why it is so protective, because it is concentrated into the brain and into the mitochondria, is that you need an excipient. And these different excipients listed there, DMSO, tween, cyclodextrins, and ethanol, have all been used with melatonin to make it into an IV formulation. We strongly believe that we need to give it IV. We need to get the therapeutic levels at the bottom here of 15 to 30 milligrams per liter in as quickly as possible to counteract those free radicals and stop that cascade of injury happening. We've translated this into what we think is the appropriate dose of 20 milligrams per kilo melatonin per 24 hours, and have done intensive pharmacokinetic studies in the piglet to see that this is what leads to a therapeutic level. So we published in Brain this very highly protective melatonin augmenting hypothermic neuroprotection, and obtained quite quickly an orphan drug designation in 2014. So as you know, orphan drug designation is for a life-threatening or chronically debilitating condition that occurs in less than 5 in 10,000 people in the EU. We then were lucky to have an industrial partner and licensed the orphan drug to them, and they developed a formulation, us thinking that probably we shouldn't be using ethanol in babies, that involved cyclodextrin. We then moved on and worked on melatonin, defining the dose and the neuroprotective efficacy. But then in 2021, the industrial partner pulled out. We also realized at this point that actually cyclodextrin was not something that we could be using as an excipient and take a therapy into babies. The EMA say it's contraindicated in newborns due to the scarcity of safety data. At that same time, having been in a rabbit hole for a long time, we suddenly woke up to actually the data that was sitting on our desks, that actually when we combine melatonin with ethanol excipient, in the piglet and fetal sheep, Alistair Gunn's work has shown this as well, we actually see augmented protection. We went to the MHRA and they gave us some sound advice, and we also went back and looked at all the preclinical data that has ever been looked at in melatonin and saw that melatonin is a goer. This is very briefly this low, what I thought was a low-lying paper looking at high-dose melatonin with ethanol in 3 groups: control, which is cooling, control and ethanol, control melatonin and ethanol. You can see that the EEG recovery was brisk and apparent in the green on the upper right with melatonin ethanol, but that we were getting partial recovery with ethanol by itself. So too with the cell death, reduced cell death with ethanol, and better oligodendrocyte recovery with ethanol. MHRA said to us, yes, you can use your MRS lactate to NAA, but you need to keep your blood alcohol levels below the FDA threshold of 0.25 grams per liter. We went away and we worked out pharmacokinetics and indeed could keep our ethanol level low, below the FDA guidance, and we could also keep our melatonin within the therapeutic range. Then my lovely PhD, Ray Pang, did this meta-analysis of all the preclinical studies of over 100 animals, and again saw in rodent, sheep, piglet, different animal models with and without cooling, melatonin had a significant benefit. So I spent a bit of time looking at how scant the information on excipients for neonates is. What we are dealing with is this publication, you may know of others, Committee of Drugs, some 40 years ago, suggesting that the amount of ethanol contained in any preparation should not produce a blood concentration more than 0.25 grams per liter after a single dose. In 2014, suggested that these are safety standards, and there's really very little scientific evidence. MHRA was supportive and have supported us taking this into an early phase clinical trial because the significant weight of benefit of melatonin overrides a potential theoretical risk of short exposure to ethanol. We are continuing, and we very much hope we can take this into an early phase 1 dose escalation study, the Acumen study, acute high dose melatonin for encephalopathy of the newborn. We have obtained FDA ODD for melatonin ethanol as both actives earlier this year, and we are in discussions with EMA to obtain ODD for melatonin with ethanol as an adjuvant excipient. But key points, I feel time has been lost. We really need support for academics to navigate the regulatory hurdles and pitfalls. We need more knowledge on the use of excipients in neonates. And in this situation, translation being dependent on an industrial partner was insecure. It's been a great journey to meet colleagues who have suggested there is support out there, Connect for Children, for example, and the MHRA are running an innovative licensing and access pathway to accelerate the time to market, etc. I just want to leave with a suggestion, and that is the Stroke Therapy Academic Industry Roundtable in this very well-known publications of all the failed treatments that have gone into stroke therapy, and the advice to use a stair scale recommendation for preclinical testing, that we should consider a clinical trial only if high levels of experimental efficacy and a diverse body of evidence supports its clinical application. And should we have an equivalent STAIR guideline for the translation of preclinical neonatal encephalopathy studies into early phase clinical trials? And I'll leave the table here with the stroke guidelines and whether this is something we should be working on together to have neonatal translational equivalent guidelines. So thank you very much for listening. I look forward to the discussions, and just to say that all this work is done by my preclinical team at UCL, and thanks to my funders. Thank you.
Good morning. Nikki, you're a hard act to follow as usual. Let's see. So I've been given the task of talking about challenges of neonatal neuroprotection trials. And I thought, I have no disclosures. I thought I would start with this pie chart that was adapted from the Lancet in a very recent article, just to show causes of neonatal death. And neonatal death is about 46% of under 5 mortality. And if you look at this, you can see preterm birth complications make up the largest section, and interpartum related events, which basically is HIE is what we're talking about, makes up 24%. So this is a very significant problem that if we could just figure out a way to prevent and or cure it, we would make a huge impact on neonatal health. I next wanted to just say something that was very obvious, but I think is worth saying nonetheless. Neonatal survival is improving, continues to improve, but outcomes for survivors are not improving. And so if you look at prematurity now, over 95% of preterm babies survive, but still almost 50% of them have neurologic injury or neurodevelopmental issues. And similarly with HIE, we've improved survival of these babies with cooling. But as Nikki mentioned, many of them still have neuromotor or neuropsychological problems. And the other thing is that the cost of neonatal care and innovation is minimal compared to the cost or the burdens borne because of adverse long-term outcomes. And so if the outcome for a neonate is poor for the rest of that child's life, they can't contribute fully to society. And not only that, but it costs quite a bit to care for them. So I think the last paper that I saw that actually looked at this in dollars was in 2003, and it was about $1.3 million to take care of one child with CP for their lifetime. So multiply that now by whatever the number is to get to 2022 dollars. It's very significant. So it's trite, but it's true. Babies are our future, and the determinants of health outcomes for adults is one branch of thought. And also just it's important to keep in mind that there's transgenerational effects of neonatal health. So that was my soapbox. And now I'll talk about getting there from here. So if you start out, your question is, what is X, whatever X is it therapeutic? So for Nikki, it might be melatonin. For me, it was EPO. So then what animal models do you work with to show whether or not it's therapeutic? The work goes into figuring out the mechanism of action, what dose is most effective, how many doses, how often, what's the appropriate route of administration, Safety studies need to be done both long and short term. And then how does the effective dose in rats or larger animal models scale to humans? And then which humans, of course, are you talking about preemies, or are you talking about term babies with HIE or other populations like stroke or IVH? So then there's the IND that you need to get for study in humans. You need to put it on clinicaltrials.gov. You need to get funding, and then you can start doing your phase 1, 2, and 3 trials. And then, of course, it's important to follow this up. So that's like the whole paradigm. So then the question is, are we using the right preclinical models? And factors to consider are brain anatomy. So for example, a rat has about 11 to 12% white matter compared to a human that has about 50% white matter compared to gray matter. And so if you're studying white matter disease like preterm brain injury, the rat is not a good model. So you need to think about preterm versus term brain injury, whether it's an acute injury or chronic or intermittent. And most of our animal models are acute injury, right? It's very difficult to get a model of chronic brain injury. And then the timing of development is important. And then there needs to be rigor and reproducibility. So just to illustrate this, if you're thinking about brain, there's a tremendous amount of development that occurs between 23 weeks when some of our babies are born to a full-term brain, as you can see there. And the complexity of the brain increases tremendously over this period. And similarly, the cerebellum develops in a similar way during these weeks in the NICU. And this is a slide that Steve Abman gave me from a recent Lancet commission that we were all out in London meeting. And he made the point that persistent pulmonary hypertension is quite different, whether you're talking about a preterm baby or a preterm baby who is now grown into BPD with pulmonary hypertension and BPD. So in preparing for human trials, you have a necessity of adequate preclinical data. And so these are basically the STAIR criteria that Nikki was talking about. So evaluating a candidate drug in multiple animal models, a dose response over a reasonable time window. You need appropriate blinded physiologic studies, histological and functional outcomes with prolonged survival. Both sexes need to be studied, and it needs to be replicated in multiple laboratories and industry, and both positive and negative studies reported, because as we all know, it's much easier to get a positive study published than a negative study. So the next step is thinking about the drug itself. And if you're using a repurposed drug like azithromycin, melatonin, EPO, it's much easier to start with because it's FDA approved and there's some safety information that's already available. However, most of these drugs are not FDA approved for babies. And so you do need to get investigational new drug applications or an IND from the FDA. Again, it needs to be published on clinicaltrials.gov. You need IRB approval, you need funding, and oh, I have IRB approval twice on there. I just noticed that. Anyway, this process can take years, many years. Once you get that and you're starting to run a clinical trial, these are some of the important steps. So if it's a multi-center trial, you need to do site visits before the startup of that study. And at that startup study visit, you need to go to the lab, make sure they have everything they need for storing samples at -80. Make sure that they can actually collect the samples that you need. Make sure pharmacy is open 7 days a week so you can enroll patients. Just all the details you need to check. Then there's project monitoring and tracking. And that graph there is from the HEAL trial. The straight red line is our projected enrollment, and the wiggly red line is our actual enrollment. So we were quite fortunate to enroll right on schedule. But then also there's monitoring of like the accuracy of the documentation. So you need to look at the chart itself and compare that to what the research records show and make sure that's all accurate. You need to monitor serious adverse events. There's a DSMB that's involved that meets twice a year to review all of the serious adverse events and make sure that it's safe to continue. Deviations and violations are collected and checked on. Maintaining research certifications is important. So for research studies, the Bailey exam is more rigorous than a clinical Bailey exam. And so we certify the Bailey practitioners once a year to make sure that there isn't any drift. And then cheerleading is huge. So one of the big factors of having a successful study is cheerleading at all levels. So the PI needs to be out there. The site PIs need to be cheerleaders, and same thing with the research coordinators, because it takes a whole team to make this kind of study happen. And the bedside nurses need to be involved and need to be excited about what the study is accomplishing. So for phase 1 and 2 studies, these are small studies that look at safety, pharmacokinetics, and the possibility of benefit, but there's not enough patients there to actually realistically say whether there's benefit. But these studies are important for determining which studies should move forward to phase 3 trials. And as Nikki mentioned, sensitive and specific biomarkers are needed. She already talked about MRI and MRS, but also plasma or urine biomarkers can be considered. And then there's other things like, for example, babies with HIE naturally have a fall in body temperature. And so these babies require less cooling to maintain a temperature of 33.5 degrees. So if you look at the blanket cooling temperature, that's actually prognostic. And some people in my lab have recently published this. So the data that's collected from these phase 1 and 2 trials inform the phase 3 trial. So when you get to the phase 3 trial, which is all about efficacy and safety, how do you pick the sites? How do you make sure that each enrolling site has adequate numbers of patients that and whether or not they're capable of enrolling those patients? You need a pharmacy. IDS is the investigational drug service pharmacy that's available 24/7. You need sample storage availability, follow-up availability, and follow-up rates. And you need to go through each part of the protocol and make sure it can be done. So I did a quick search on clinicaltrials.gov, and I just put in the word neonate. And there were 10,546 studies that came up. Of those, half were completed. Of those, 10% were phase 3 trials. 209 were industry funded. Of those, about half were completed. And 81 were NIH funded, and 25 had published results. So that's pretty dismal, right? I mean, we need to do better than this. And the other point to be made, I think, is just the tremendous amount of waste of resources that go into this. And resources include, there's only so many HIE babies that are available to be enrolled in a study. If they're enrolled in a study that isn't completed or isn't published, what a waste. So 42% of pediatric studies fail. And why is that? Are we failing because the drugs don't work, or because we aren't doing the trials correctly, or some combination of those? And the definition of pediatric studies failing it varies. So one definition is the study was successfully completed, but the results weren't what we hope to find. Another definition is the study fails because lack of enrollment, or because it wasn't done properly for one reason or another. And then the other thing to keep in mind is unintended consequences. And I'll just show you a couple of examples from the PEANUT trial. So the PEANUT trial is a preterm EPO neuroprotection trial. We enrolled 941 babies at 19 sites and 30 hospitals. And babies were randomized to EPO or placebo. And we had pretty strict protocol for many things, but one of the things that the PIs could not agree on was how much to protocolize giving iron, even though they acknowledged that if you give EPO, iron is important. So here's some of the results. We definitely showed that more EPO-treated babies remain transfusion-free, and babies who were transfused had less volume, less exposure to donors, and less actual transfusions. And we did give iron, and you can see on the right-hand side, the babies in red are the EPO babies and blue are placebo babies. The EPO babies got more iron than the placebo babies, but it turns out that they didn't get enough. And this, so just going back, you can see that the average peak amount of iron was around 4.5 milligrams per kilo. And the recommended dose, if you're on EPO, is 6 to 8 milligrams per kilo. So these kids got iron, but not enough. We did have the sites measure ferritin at 2 weeks and at 6 weeks and adjust the iron dose based on that. But you can see in this graph, the yellow block on the bottom indicates low ferritin at the level that actually affects neurodevelopment. And you can see that a lot of the red dots, each dot is one child enrolled. And a lot of the red dots were patients who were treated with EPO who were then iron deficient. So unintended consequences. This just summarizes the data looking at iron dosing in these kids. So these on the left-hand side, you see the 3 components of the Bailey test. So cognitive, motor, and language. And what is shown here is that the more iron you got, the better you did. And that's particularly true in the EPO-treated kids. So these babies were undertreated with iron, which affects neurodevelopment. The other issue is outcomes. And frequently, when you plan a study, you're basing your incidence and complication rates on published studies. And so when we were planning the PEANUT study, we looked at Barbara Stahl's beautiful study of complications of prematurity and used data from that study and other comparable studies showing that death or severe neurodevelopmental impairment was around 45% in this particular group of babies. But what we actually saw was that death and severe neurodevelopmental impairment was lower than predicted. So perhaps because these babies were enrolled in a study and that alone affects the care. Also, because of the entry criteria for this study. So in this study, we did not enroll every single baby. For example, if you were moribund or if alternate care was provided, comfort care, they weren't involved in the study. And those babies contribute to the death rate. And so basically, we were underpowered to show a difference because of this. Site variability is another issue, and these are a couple of examples. So these are the 19 sites laid out from 1 to 19, and you can see just a tremendous amount of variability in sedation use. So the top panel looks at opioids and or benzodiazepines. The next level in blue shows the use of opioids, and it's divided in morphine and fentanyl. And the last green panel shows the use of benzodiazepines. And we did show in this study that prolonged use of opioids or benzodiazepines adversely affect outcomes. And so you can easily imagine here, there's quite a bit of difference by site. And similarly, this is the difference in use of steroids. And so some sites used a lot of steroids, some sites used very little, and the cumulative doses varied quite a bit by site. And we all know that's another thing that affects long-term outcomes. So again, 42% of pediatric studies fail. And why is that? One reason is that they might be underpowered. The patient population was not as sick as predicted. Patients drop out early, or patients don't return for follow-up. So it's difficult to get. A lot of these centers are referral centers, and patients come from quite a distance, and sometimes they don't come back for follow-up. So it's important to pick sites that have a track record of 90% follow-up or greater. Underfunding is another reason. Average cost per patient, this is published, ranges from $40,000 to $90,000 per patient. The peanut study was 940 babies and was $10 million. So like funding is a huge issue. And then there's the regulatory burden that increases the cost because of all the work that needs to get done, filling out forms and so forth. And then the wrong endpoint can also be a reason that studies fail. So low enrollment is a big problem. I'm on the DSMB for a few studies, and enrollment is a big issue, I can tell you. And why is that? Well, people overestimate their ability to enroll patients or their patient numbers, or the numbers change. There might be competing studies at a given site. The availability of enrolling staff or pharmacy or lab can affect this. So you have a patient who's eligible, but it's a weekend, nobody comes in to consent the family. And oftentimes neonatal studies in particular are very time sensitive, right? So if you want cooling studies, they need to get in by 6 hours. The peanut trial was enrollment by 24 hours and so forth. So that's an issue. Long, incomprehensible consent forms. Consent forms vary widely from place to place. So in the peanut study, we had a site that had a consent form that was 6 pages long, and we had a site that had a 19-page consent form and everything in between. And so you can imagine that parents are not going to sit down and read a 19-page consent form. They just won't do it, and they won't enroll. So mistrust of science and medicine, I think we've seen an uptick in that recently. And people just don't trust the doctors in the same way that they used to. And this varies by racial or cultural group as well. That's also a problem because if you get low enrollment in a particular group, then the study results aren't generalizable. Acutive enrollment, I already talked a little bit about that. So the albino trial aims to enroll patients by 3 hours after birth. So you can imagine that's almost impossible, and that's reflected in their enrollment numbers. Investigator bias. There's been a couple of studies that have tried to look at which anti-seizure medication is optimal, but it turns out people really believe in whatever drug that they use. And so they won't enroll patients to use something else because they think that that's not the best thing for their patients. The critical nature of neonatal illness and the emotional burden can also be a problem. So for the HEAL trial, enrolled 500 babies with HIE at 17 sites. And a lot of the mothers were at a different hospital because the babies get transported in to a referral hospital. So consent was on the phone. The dad was often on the way, and we needed to get consent from both mother and father. That was a problem. And then the interaction between the potential enrollee and enroller is really important. So finding research coordinators or whoever the enroller is going to be needs to be empathetic and somebody that that invites trust. So what factors accompany study success? I think asking the right question is important. The right study design and statistics, the right medication at the right dose, the right inclusion criteria, the right exclusion criteria is also just as important. Enthusiastic team leaders and dedicated research coordinators, and plain language, short consents are really important. I also just wanted to mention, I have spent my my career basically looking for a silver bullet to help these babies have normal, full lives, thinking that maybe if you're born prematurely or with HIE, you're kind of behind the starting line and the goal is to get them even with everybody else. But it turns out most of brain development occurs after discharge. And what we're learning more and more is that development really depends on the family these children go home to. And maybe the silver bullet isn't something that you do in the NICU. Maybe it's early intervention and parenting classes and those kinds of interventions. So challenges, regulatory constraints. I've already talked about the consent forms, FDA and IRB. I think it's important to communicate that the risk of not doing studies is not appreciated by the groups trying to protect the vulnerable. And so, I deal with this all the time with my IRB. It's like they want every single little risk factor. It's like if you start an IV or give an IV drug, they're like, well, you have to talk about bruising and extravasation and all of these things that are important, but they put so many things in there that when you read the consent form, it's like, why would you ever consent to this study? It seems like a bad thing. And so having a balance in the consent form is important. Mistrust from families and patients is a challenge. And I've talked a little bit about DEI and just the generalizability, and then the ethics of studies. So if you don't if you have a study that isn't completed, then is it ethical to enroll patients in that study? I mean, that's something to think about as well. So it's really important to have well-designed, completed studies. And then follow-up is essential, but expensive, and who pays for that? We need linked prenatal, natal, and childhood databases so that you can have lifespan data. And then funding is always an issue. So how do we get industry to partner with us, along with NIH and other funding institutions? So possible solutions, I think, involving stakeholders more. We don't do a very good job of that in academics. So getting families and industry involved, establishing standards of care. And I showed you just a few examples from the Peanut trial, how different sites really practice medicine quite differently. And so it would be great if we had standards of care that people used. I think involving parents, nurses, APPs, the whole team is really important for a successful study. Standardizing protocols, shorter consent forms, AI modeling, I think, is going to come into its own in the next decade or so. And then involving industry. To involve industry, we need to incentivize industry. And, because they don't do it out of altruism, right? They're companies that need to make money. And then lastly, more innovative trial designs, such as adaptive designs or using real world data. So these are some of the funding agencies for neonatal studies. And thank you very much.
Hello again. Thank you to Sunny, Ann, and Nikki. Let's bring up the panel so that we can continue. Those were such excellent talks, really laying out the landscape. Everybody come on up. Laying out the landscape of the study, the science, the preclinical work that has been done and is continuing, and the clinical trials. So I'm just going to ask for each speaker to introduce themselves briefly. So we'll have the panelists who did not give a talk give us a little bit of information briefly, and then we can get to some audience questions. So take it away, Susie.
So I feel really badly. I'm the only person not sitting on a sofa. Maybe I should.
You can.
All right. I'm going to,
Yeah.
I got to come be part of I just wanted to be able to see my slide. I wasn't trying to be isolated. All right. I'm Susie McCune, a neonatologist, spent about 18 years academic at DC Children's and Hopkins, and then spent 18 years at the FDA with Jerry and Ann, and actually in my academic career, spent time with Ann. Lovely to be working with Ann again. Just as part of my regulatory, I think it was the second week that I joined the FDA. I was in the Center for Drugs, and they said, well, we have some neonatology thing over in the Center for Devices. Do you think you could go help them out with that? So that was CoolCap. So I was the reviewer for CoolCap, and just really thrilled that we were able to move forward with that. Similarly, as to how the FDA moved forward with surfactant, we talk about having at least CoolCap and surfactant, but if you go back and look at the data were not gangbusters, and they were actually difficult regulatory approvals. And so even the ones that we have hurdles on, or have overcome the hurdles on, were not easy. So sorry, I was my last 4 years at the agency. I was a director in the Office of Pediatric Therapeutics, in the Office of the Commissioner. And then about a year ago, I wanted to see what it was like in the implementation space. And so I went to the CRO, which is a clinical research organization called PPD, or Pharmaceutical Product Development. We were actually acquired a little over a year ago, or about a year ago, from Thermo Fisher Scientific. So we're part of that now. I'm thrilled to be here. Thank you for inviting me.
Oh, and do you want to go ahead and give do you have introductory remarks or?
Oh, do you want me to do
comments?
or
Yeah.
as opposed to?
Yeah.
Oh, okay, cool. Yeah, yeah. So actually, if you go to the next slide, that would be great. So I wanted to put this a little bit into context, but fabulous talks by Nikki and Sandra that really covered a lot of this and identifying, kind of where our gaps are and how we can hopefully as a community work together as the stakeholders in this space. We start with the little feet on the left. And as we heard today, working through academia to really define what's in the pipeline and how do we move that to development. At that point, the incentives are really those of the academic community. And then, who tests this in these models? We heard about these great animal models today. And HIE, Nikki talked about all the work that's been done over the past more than 40 years in this area in terms of the models, but we didn't get into some of the a lot of the preterm animal models, which is a very different disease or different diseases, and all need their own models, as we've heard about today. And who's developing those? And how predictive are they? We talked about failure going from phase 2 to phase 3. But how do we predict what goes from models into the clinical trials, and how predictive are they? Clearly, the HIE models we heard about today look really good and have been supported throughout the years. But there are a lot of areas in neonatology, even in preterm neurodevelopment, where we don't yet have the best models. And then the development of biomarkers that we heard as well. And the incentives here still are in that academic space. But then we're starting to transition to the development of assays in terms of validation of those assays, needing small volume samples. And now we're starting to move into that drug development space, which is something that the academic community does not have the support to do, has to partner with other either biotech or, as we heard about, pharma. And how do you get funding for that? And then what's necessary? You're starting to get into the regulatory requirements in terms of toxicology testing, juvenile animal testing, GLP kinds of aspects. And I used pharma just because I didn't want to put pharmaceutical or industry as a huge title in the slide. So excuse me for using the pharma acronym. But, in that, so a lot of academics then start to work, especially with the biotech community, but those are also small groups of folks that we heard about trying to undertake all of these efforts, how to commercialize the product that we heard today about with excipients and large batch scale up with GLP. Now we're starting to get into those financial needs to be able to do these studies, and who's paying for this. And, what's interesting is while industry in general, has more money to support this, from a biotech perspective, a lot of the smaller biotech firms are actually looking to venture capital to be able to support this. And the problem is venture capital wants a assurance that the regulatory pathway is well established, that the endpoints are well established, and that we know how we're going to be able to get from A to Z with some certainty. And unfortunately, in the neonatal space, that's really hard. And so then we start talking about what Sandra and Nikki talked about, do we need to do first in human studies in healthy volunteers, and what do those phase 1 studies look like? Then going on to the phase 1b, 2, 3 trials and clinically meaningful endpoints, the site engagement we heard about, recruitment, retention. Once again, we're starting now to get back into the academic community and what's in it for the academic community. And then all those regulatory requirements we heard about in terms of data standards, data analysis submissions to the agencies, and just a few of the agencies. I couldn't put all of them on the slide. And ultimately getting back to benefiting the patient. And one of the things that is not included here, and we need to figure out exactly where to start including patient-parent input in protocols and nursing input in protocols. Because by the time we get to that bottom row where we're starting to get into the phase 1-2 trials, it's too late. The protocols are set in stone. Nobody wants to change them. We need to be having this conversation and thinking about what those protocols look like in that top level. And we're not doing that yet. So I apologize for speaking too long. I'm very passionate about this because I think we all need to work together to be able to get it done. And we've had great examples this morning in the talks about, how to be successful in this space. And even when you are as successful as Nikki and Sandra are in this space, how long it takes, how frustrating it is, and how we've just not been able to be as successful as we'd like to be. But thank you.
Thank you, Susie. And now we'll hear from Jill Beck. Please introduce yourself and you can give remarks.
Hello, I am very honored to be here amongst all these brilliant minds. So I thank you for inviting a nurse to be a voice and to speak up about some of the challenges that we face at the bedside, trying to put all of this research into action. So INC just completed a paper getting the ideas and the suggestions of nurses and how they feel about research. And overall, we love research. We are very involved in research. We support research. We know that research is the endpoint to improve our patient outcomes, right? So we want to support it, but we face a lot of challenges. So we face just daily bedside care, time constraints. We all know that, we talk about being in the trenches of research. Well, there's trenches in the bedside, and those trenches are getting worse and worse with just the climate of healthcare now. So those time constraints and everything, and being a patient advocate and a parent advocate takes a lot of time. But we want to be supportive. But we're the ones that are carrying out daily blood draws or gathering the data, inputting everything into the electronic medical record, so then it can all be extrapolated for your research studies. But oftentimes I feel nurses feel like there's a challenge, assimilating that information and how does that affect the study, and how is that going to affect the outcome and what you're measuring. So I think it's important that we're included, just as you had mentioned, from the very beginning, because we're the ones that are actually putting everything into place or talking to the parents. And that's really important that we're involved from the bottom, because, if we're not included and we don't have that, you don't have the buy-in, then the success of that trial really might not go well, because you don't have what we can offer, or you don't have the cheerleaders like you mentioned, that we want to do this because we know that this is going to help our babies. There's plenty of trials that I've been involved in where I work, where we're gathering data, we're doing these blood draws, but you don't really know what's the endpoint, what are our outcome measures here, and it just starts to become a task. So I really think that if you want to have active participation and to have successful outcomes, and we are a voice to the parents. Deb said the other day that we're the trust point, and we are the trust point because we're the ones that are trying to explain stuff to parents. The docs walk away, and then I'm a translator, and this is what's going on. And they want to know, what do you think? What have you seen? And they really respect and trust us. I think we've been, I don't know why these aren't going, but this one? Oh, this one. We spend 85% of our day at the bedside. We're with the parents all the time. So, it's important that, we understand the research, we understand what's going on, so we can explain it effectively, so the parents can make that informed consent. And then throughout the whole meeting, we've always talked about site variability, and we've talked about just all of the lack of standardization of care. And this middle picture with the little baby kills me in a neuroprotective way, because that's horrible positioning. And that's why I put it there, because it just goes to show, we think we're doing some great work with neuroprotection, but every center is different, right? And every center has availability of products, and some might not have devices that are helpful, some do. And this all can affect any kind of study that we're doing on neuroprotection. These are creating all these challenges, to put research and to translate that research into bedside practice. So, I don't want to keep talking, but, we really can bring a lot of insight into the feasibility and operational issues to why studies aren't being successful and not failing, just not being successful, and you're not getting the outcomes that you want. So I feel that nurses, if we're starting from the bottom, we can bridge that gap together. And if we're involved in early planning and study design, it's really going to be able to bring our stakeholders together and effectively improve all of the outcomes, and why we're here, to improve our babies' lives.
Thank you, Jill. I'll turn it over to Betsy.
Thank you. My name is Betsy Pilon. I have the amazing opportunity to serve as Executive Director for Hope for HIE, which is the world's largest patient support and advocacy organization connecting families. As I mentioned yesterday in my video intros, I am also a mom and entered this world 10 years ago. It's an interesting time to enter, because as the presenters shared, 10 years ago, it was a very different place than where we are today. And Max was really in kind of that cusp era of cooling becoming, standard of care. And even, the multi-center whole body study long-term outcome paper came out that week that we got home from the NICU. And I still have that clipping from the Detroit News from my mom. So, my role with Hope for HIE is to elevate the voices of these families. And we are now connecting over 8,000 worldwide. And we have a whole lot more to connect when you crunch the numbers. But we are very, very fortunate in this collective brain trust to have families that were part of the Toby trials, that were part of the original multi-center, that are part of the head cooling, that were a part of the HEAL study, that are, from all of this lived experience. And we're aware of the challenges. We're aware of, of the pitfalls and the opportunities to be a better supportive partner from the beginning and take that lived knowledge. Our families in particular, I feel like I sit in so many conferences where we talk about how sad HIE is, and it's true. But, our organizational name is Hope for HIE for a reason. And so we're always trying to bring whatever the outcome is, a glimmer of hope in some way, and to, some very tragic situations, for our lost families. And one of our board of director members I talked to last night, she lost her son, and he passed away at 6 weeks old. She has been serving on our board and grew our lost group. And the hope that she has every day and why she gives back to our organization is that people like this panel are moving forward and going to help and change and see what the trajectory is. And it should be from 10 years from this point, something completely different as well. So, we've done, we've been able to partner with, some different studies, some different collaborations and research, one of which I think has been, I like to tout as the gold standard of PCORI. We've been a proud member of the Neonatal Seizure Registry Project since 2019. And the type of engagement, the type of conversations we have within, that space is really, I think, tackling a lot of the barriers that we talk about today with, these, you have to do these long-term outcomes. And we see that, we have, like I said, we have data, this anecdotal data of 12 plus years of information sharing. And we can see these trends. We see these kids coming back. We see the, sign-offs at age 2 and 3 and the return with seizure onset, with learning and attention issues and all these things and the full impact. And so we know just how important it is to keep families involved. And I talked a little bit about yesterday, the concept of halo of support and, what a benefit is to partnering with a patient advocacy and support organization like ours. And Rare does it, I think, the best right now. There are really so many things that we can take from the Rare space and the people that are just digging in there to move things forward. And really looking at how you can partner, how we can reduce attrition, how we can make sure messaging is on point, messaging along the spectrum. I happen to have a communication background in marketing with healthcare and with, obviously understanding acutely what our families talk about. Looking at, and I know Yvonne, reached out at the end of HEAL and said, we're getting ready to publish this. And we know that, and she said, we're concerned about how the families are going to feel. And I was able then to prep our, prep on the back end for when it posted out to the world with the results and have that, we had our social worker ready to facilitate conversations because these families are very invested. Everyone, again, that hope they're carrying that this is something that is not only going to benefit their child, but hopefully someone else, just like the pioneer parents of cooling. And, we benefited directly from that. So I think, we're able to we're able to start from the very beginning. And we're doing so with all sorts of different key stakeholders, with academic, non-academic, bioscience companies as well. I'm excited. The future of HIE is hopeful. I've probably had 10 different research groups and all around the world reach out to us this year and at different phases of where they are in development and say, hey, let's talk about how we can work together. And so that, that is becoming the standard and it needs to continue. Again, I think, the Rare side of things has built and developed so many best practices that Neonatal absolutely can take advantage of. There's no need to recreate the wheel. So taking those templated formats, and people are so eager to mentor and help. And I feel like I've been very fortunate to, especially in the rare epilepsy space where HIE is a cause of many of the rare epilepsies and leading cause. Those are the people that have been knee deep in the science because epilepsy is so complicated. So, and the movement there is great. But, I always ask this of the, the LGS foundations of the world and saying, well, okay, HIE is a leading cause. What about backing up to, as we talked about prevention, if we can decrease the impact of HIE, you're decreasing all of those other things. We're talking about kids living their life to the fullest and living, having these great potential. Now there's a lot of, wonderful things that have been done with accessibility, and that continues to move forward, but not at, the levels that obviously, gives every child a chance to thrive and reach their individual potential. But I want to thank you all for having me here, for listening to the voices of patients and families and making sure that we are all, at the same table. And because that's, I'm a strong believer, and I know this is why Inc. is set up this way, that when you get all the right people around the same table, you're going to get the best results. So thank you.
So thank you so much to all the excellent speakers and panelists. I have a lot of questions for this panel, but I was wondering first if Anne has any specific things you wanted to follow up on first. No, okay. Heard a lot of barriers and challenges, and so I'm wondering if anyone on the panel could address one of Dr. Robertson's comments, which is that academics need support to be able to translate from the lab into the real drug development space. Is there anyone who would like to sort of take on, how that might, where we might be able to support them? Dr. McCune.
Jerry is looking at me like I have answers to this. And I don't know that I have answers, except being able to be part of a community like this to know where these resources might lie. And, as I pointed out in the slide, and one of the things from an academic perspective, in terms of supporting these kinds of translational efforts, it's clear to get grants, to get support, to publish research papers. That's the coin of the realm from an academic perspective. The problem is when you start getting into industry type trials, there's really not the support from an academic perspective for career development for academicians for that to be seen as a tremendous positive from career development. So I think there has to be a change in the recognition from an academic perspective, the value of these industry or drug development trials. I think there have to be connections between, there are a number of academic folks that then essentially create their own biotechs, but then how do the biotechs then get to industry or CRO partners to be able to implement these trials? Where, how do we connect those dots? And I think that Inc. is a great opportunity in this space to be able to make that connection.
Thank you.
I'll just add to that too. I think, just a quick addition. I think things like Inc., I completely agree. I think there's such silos. So I think it's funny that I'm here as the regulatory representative because I spent, nearly 20 years in academia and just recently transitioned to the FDA. And I'm still learning about everything that happens in the FDA. And Sunny and I were just talking about this yesterday, that even with Inc., it's a huge body and doing a lot of great work. And I think, not everybody in some of these specific disease areas knows about it. So I think meetings like this, I think having folks that kind of bridge the two realms. Jerry was speaking at a recent conference at the Children's Hospital Neonatal Children's Hospital Database Consortium. So I think the more bridging there is to understand, what the different perspectives from the regulators and panels like this is where those silos might be broken down.
Can you hear me?
Mhm.
That question is a very important one. With the second drug that I tried to develop, intravenous ibuprofen, it was very clear that at some point I had to give this in to a drug company because of many reasons. Why? One of the phase 3 trial needed millions of dollars. We were part of the PPRU, and that I think gave an academic recognition to the work that we were trying to do, that it was still an academic initiative. But at the same time, now the funding was shifted to the drug company. What was the price? I have to give them, this is your patent, this is yours. The other reason why it was very important, somebody has to do the formulation, somebody has to sell it, somebody has to do that. You can't do that from a university. So there is no way, as far as I know, as I can see, that you can manage to keep it your own. At some point, you have to give it to the people who can make sure that the standards of the FDA and the EMA and the regulatory bodies are maintained. Good formulation, safe formulations, and all of that stuff. So I think the challenge for us is how do we slowly get into that? The first phase was of this intravenous ibuprofen project was published in 1995. Phase 1, 2, 3, and 4. The FDA finally gave approval in 2006 after the NDA. And that gives, I think, Dr. Jewell had felt some discouragement that it takes so forever. No, it isn't forever. This was about 11 years. And so there's hope. I mean, it takes time, but we need to have that to make sure that these drugs are safe and the proper studies are done. And
Thank
so
you.
I think we can manage
Mm.
to still keep the academic component of it, at the same time, nicely give it to the drug company to sell it, to advertise it, to do all the necessary maintenance of quality control.
Thank you, Dr. Aranda.
And just to add, Jack, that's why I went to the clinical research organization space, was to try to figure out how to link between the academic and the regulatory.
The other one, just a couple of comments for Dr. Nicholson and Sandra. First thing is that I was impressed with Dr. Nicholson's presentation, particularly on the secondary energy failure in HIE. What people had really not done is how to accelerate the secondary, how to prevent it, and how to make the mitochondrial energy system functioning. For instance, nobody has asked that question. If you, for instance, accelerate or give something for component 2, ubiquinone or coenzyme Q10, right? And make increased production of ATP at those very critical time. What would happen? I mean, no one has really asked that. They keep trying to prevent it, but the energy failure is coming. What you need to do is to try to see whether you can make more ATP from that, or could you do it from component 4 by giving ADP to try to make more ability to make ATP. I don't know those questions. I'm not a neuroscientist, but these are some of the challenges: asking the right questions, and then trying to make the appropriate protocols. And again, the question of the melatonin ethanol is a fascinating one.
All very fascinating.
But have they asked the questions about what is going on in here? Is there a change in receptor binding affinities? I mean, just no one has done those things. I mean, there are many questions before we try to get it down into those poor newborn babies that need these drugs.
Right. Thank you.
Yeah.
Thank you. Is there any thought, and I see that we have another question in the room, but is there any understanding of what the ethanol might be doing in that model?
Yes. Can I just answer one of your points first about complex 4 of the electron transport chain, which you have asked about? In fact, melatonin is going in there, preventing ATP loss and preserving the electron transport chain. So we do in the lab look all the time at what is happening to the energy failure. And that's, I think, what makes it so translational. In terms of the ethanol, I took out the slide that actually described so eloquently the preclinical studies in rodents, albeit, but there are about half a dozen of them that show a very low dose of infused ethanol below 0.5 grams per liter is protective in 4 fronts. It's anti-apoptotic, it's free radical scavenging, it acts as a fuel, and I think also provides a redox change. So it is very well known there that ethanol in an acute infusion, low dose after hypoxia ischemia is protective. But, you know,
Wow,
it's difficult.
that's
to
incredible.
change,
Yeah.
you know,
That's
what everyone thinks about ethanol.
but science. You know, you have science. So go ahead and please introduce yourself.
Hi, my name is Sonia Wong. I'm a pediatric neurologist with a subspecialty in epilepsy. And I can relate to everything that every single person has spoken on. And I would love to be able to ask, is there opportunity for mentorship for people who want to engage in a research space? Because as an academic, but also a practicing physician, there are many pressures that, I mean, we have a 6-month wait list to see a pediatric neurologist, but we are the ones who follow the patients all the way through 18, 20 years old. I have I am from University of Minnesota, so we had the largest nursing strike. And so I couldn't enroll for almost a month and a half. So, and I've put in IND applications, not knowing what to do, just went online and said, hey, I think this is what I'm supposed to do. So I'd like to just propose and offer. I put in an IV melatonin application and put on full clinical hold and said, okay, this is too overwhelming. Let me just put it on the side and do my seizure trials. So it would be wonderful to see if there's an opportunity for mentorship for people who want to explore the space, but at the same time, understand that we can't do it all. I do think that there is space for development because I have a whole team of pharmacologists who said they can create the IV melatonin, they can do stability and sterility testing, but it feels like we're siloed and flying by the seat of our pants. So mentorship.
Yeah, so absolutely. I think we encounter a lot of reach outs and obviously there's our guidances that are available. And, I think that's part, our role as the neonatology program within the FDA to be a resource to point you in the right direction. And obviously, expect that at some of these universities, there's regulatory guidance and consultants, but I understand, in this space in particular, there's all these challenges that we talked about today that being partnered obviously with somebody who's been through the process and put in a successful IND in this space and those kinds of things. So I think there's opportunity for guidance and pointing folks in the right direction from the FDA side, and I'm sure the same across the pond. And then obviously seeking out academic mentors that have gone through the process successfully is going to be key there as well.
I think TCAN is taking that on. They've tried to connect, solicit
TCAN?
mentors and then mentees, and they try and match them together. So that's a relatively new program that I know I've been involved in. So
What is TCAN?
Oh, I can't remember what it stands for, but it's a group through the AAP that
The section for
for
neonatal perinatal
neonatology.
medicine.
Yeah, sorry about that.
Trainees and early career. Yes. Yeah, academic neonatologists, something like that. So I'm sure they would welcome a neurologist as well.
Good morning, everyone. Thank you for those wonderful presentations. I will declare that I'm not a physician. I'm a clinical pharmacist now, or a clinical pharmacologist. And so our mission here at INC is to develop drug products suitable for neonates. One of the things that I think we should be focusing on is the right kind of pediatric formulations. For any clinical trial to be successful, you have to be able to give the right dose and the right drug, but it should go a little bit further in the right kind of formulation. As Dr. Aranda just mentioned, IV ibuprofen is a wonderful example, but we have problems with IV melatonin. And so those are examples of pediatric formulations. So I think in general, in order to have successful neonatal clinical trials and have them from failing, we need to find the appropriate pediatric formulations that can be given to neonates, starting from premature neonates to however old, 2 months, 3 months old. And there are many roadblocks along the way. Every formulation is not an IV formulation. Many formulations are oral formulations. So we need to understand how to administer those drugs to babies who have NG tubes, G tubes, etc. Not only that, but once the drug is administered, what is its impact on pharmacokinetics, pharmacodynamics? That is going to lead us to the ultimate endpoints of safety and efficacy that we are looking for. And I think our discussions should involve some more of pediatric formulations in general that we can use appropriately in neonates. And as a disclaimer, I do work at the FDA now, but like Anne, until 2 years ago, I was a faculty at University of Michigan and a neonatal practitioner who was at the bedside for almost 32 years. And this was my constant struggle. How do I get this drug into this baby in whatever format it is? So my humble request, let's also address pediatric formulations. Thank you.
And we certainly heard that from Dr. Robertson about excipients and formulations and how little is known. And so I might be hearing kind of an action item, potential action item for INC to consider. Did you have a question, Dr. Pileggi?
Actually, more of a comment. Antonello Pileggi from NICHD, NIH. Regarding the workforce and how to get a more prepared workforce. There are great opportunities. There are a lot of training grants that for which the training can be shaped as part of enterprises, as part of a consortia. So there are opportunities to bring a workforce to capacity with a grassroots type of effort or an academic type of input. We need a workforce that is more entrepreneurial in a certain way to kind of get a perspective, but also transdisciplinary in order to get the input from the pharmacologists, the neonatologists, etc. I think that there are opportunities out there, and NIH, at least that's the NIH perspective I can bring here to the table, is trying to incentivize that. So come and look at me. I can talk to you.
Thank you. So there was a question online. It was more of a comment, but from Dr. Prakash Shah, and so hi to Dr. Shah. He was asking about the discussion about failed trials, and he wanted you to comment a little bit more on, whether it's correct to call something failed. If it failed, it's, if the null hypothesis was rejected, is it truly a failed trial, or is it just that the investigator didn't get the result that they expected?
That's why I went through the different definitions. So I think, in my view, a failed trial is a trial that doesn't get completed, or it's the wrong question being asked, or the statistics weren't right, or, that in my mind is a failed trial. I think you can have a successful trial like both HEAL and PNET that didn't show what we were hoping to show. So I think those trials are successes. They got completed. Exactly.
Right.
And in some of the reporting that we've done from the FDA, some of it is thinking about, especially in the PREA, the Pediatric Research Equity Act, where there are requirements to do, and this is outside of the neonatology arena, requirements to do pediatric studies for drugs that have been approved in adults for an indication. And what we were looking at is, were those products then, when they were studied in pediatrics, then approved for pediatrics? And that was the failure was, here was a drug that was approved in adults that failed to show efficacy in the pediatric population. And then that gets into a different question of, why did it fail? Is it a different disease? Did we not use the right dose? Did we not, study the right population? So, so there's, I think you can look at failure many different ways. And in the literature, I think you have to parse out what the definition is.
Yeah. And I'll just add, that's why on my slide, I failed in quotes, because I don't like that either, in again, different perspectives, right? So from a regulatory perspective, we say that those are failed because they didn't meet an endpoint that could be used for regulatory decision-making and an improved therapeutic option. And I remember the HEAL meeting when you guys discussed the results to the whole investigator team. We said, this is not a failed trial because we answered the question. It was a successful trial. It was completed. We answered the question. We knew that it didn't help and it may be harmful. And, from an academic perspective, it was a big success because it answered the question.
Mhm.
So it, again, just highlights the different perspectives when we look at these issues.
So I have yeah. go
I was
ahead.
just going to make a quick comment. And I think from the family side that, you know, hearing and I mean, families that have been enrolled from the beginning with HEAL in particular, and tracking their experience through that, of course, you know, people, you know, from our side, we're always going to hope that there's something that's showing a positive benefit. But I think, you know, the conversation was really after the results were published was this is such important work that will drive future advancement, that people felt good about that they are contributing to, you know, to something that was not done before, which, you know, and I say this every day with our community, like if we're moving something forward, it's a win. And so whether that shows what we think it's going to, or what we hope it's going to, if it's something forward, it's
Right.
better than it was yesterday.
And if you put that to rest, then you can move on and look for, novel ideas, or go through that list of potential candidates and find the next best candidate to focus on.
I think one really important thing to point out is just the importance of phase 3 trials. There was a bunch of phase 2 trials for EPO that showed benefit, and there was meta-analyses that put all of those together. And sure, it was the greatest thing since sliced bread, but when you do a phase 3 trial and really look, it wasn't successful.
Right.
And just to address your question, I mean, so the volume of distribution of EPO is different in babies than it is in adults. There's so many differences, both pharmacologically and also, when you're born preterm, like whole cell populations in your brain aren't born yet.
Right.
It's really a different animal.
Absolutely, which is why in many of these cases, the FDA is not able to extrapolate from adults to, especially to neonates, because the diseases are very different. And you're absolutely right. The dose is different and the safety is different. So all of those things that really require those phase 3 trials, which is very hard because the community, they're very expensive, and the community wants to just be able to go with the phase 2 data. And I think it's really important what you point out about the phase 3 trials. Absolutely.
Yeah. But then again, I think it's important to point out the risk of not doing the studies, right? Then nothing
Right.
moves forward.
So I'm just going to say we're going to take at least another extra 5 minutes into the coffee break so that we can get to some more of the questions that apparently Dr. Davis has, and then I have for the panel.
Great, thanks. Great talks. I think it really highlights that the challenge with newborns of animal models that may or may not simulate what goes on. I've done that, spent 20 years working on animal models, not finding that it works the same when you do get it into the babies. And then Sunny, I thought your talk was great. One of the things I was fascinated with is, with your data on iron, we all think of iron as a pro-oxidant. We don't want to really give babies iron because we're worried about, unintended consequences of that too. And so, even though, and you're right, sometimes you have one bite at the apple, and yet you're trying to figure out what the best dose is. And then you go after your trial's over and go back and say, well, some of the ferritin levels were lower. Maybe we weren't using enough iron. Maybe that's why the drug wasn't as effective. Maybe the dose we picked, and we did it based on all the other phase 2 trials. Maybe it wasn't high enough, or it was too high. So those are really the challenges built into exactly what you're saying. And I don't know how to get around those except doing more of those phase 2, looking at more dose ranging studies, looking at iron supplementation and things before you get into the phase 3 trial.
Yeah, it's well known that you need iron with EPO, but the bias of neonatologists is exactly what you started with, is like, oh, iron's evil. And when I was a fellow, the rule of thumb was not to give iron until babies were 2 months old or 2 kilos, because it was so dangerous. But it turns out, you need iron for brain development and for cell division and myelination and dendritogenesis and et cetera, et cetera. So, yeah, point well taken.
So,
Oh, I have one, sorry.
yeah, no.
Just, I think it's really important to do multiple animal models because, so for example, there's a lot of data in rodents that EPO is anti-inflammatory, and you measure different cytokines and show, you know, if you give EPO, they're decreased after brain injury. It's not true in humans. I mean, it's just not true. Like we looked at it, we did biomarkers both in HEAL and peanut, and EPO does not affect inflammatory mediators. And so different animals function, you know, differently.
Right.
Babies
The
aren't
animals only get you
rats.
so far. So I wanted to address something, or for the panel actually to address the issue of efficiency and resources. And, you know, with respect to the standardization, and, you know, as an FDA person, we often see pushback at the protocol level about standardizing practices. And we know that this is important for clinical trials to be able to show whether the intervention is truly having an impact. And so I would like to start at the end with Jill and just talk about where do you see, you know, sort of low hanging fruit? I mean, we talked about the challenges, but where can clinicians and investigators and families and nurses have an impact in terms of pushing for standardized practices in care when the best practices are understood, and also in clinical trials so that we don't waste resources and babies?
That's an excellent question. And it's something I think every nurse is challenged by. Somebody said last night, you know, you get 100 neonatologists in the room and you're going to have 200 answers. And, you know, it depends on who's on service, what we're going to do, what we're not going to do. And it is very frustrating, and it does affect standardized care. And I think it should be important to capture standardized care practices that we already have in place. So we now are doing golden hour. And that's really transformed how we approach admissions of premature infants, right? So it's given us great outcomes with thermoregulation, IVH risk reduction, so that time is standardized, but then there's so many other multifaceted areas that aren't standardized, right? So humidity, you go to different units, everybody does something different for humidity. So I think if we can try to standardize our practices for what's already kind of packaged nicely with us right now. So your golden hour, all of our HIE admission babies, you know, that's standardized care. This is what we do. That I think if we can capture that and take advantage of those almost standardized practices and focus on that, then that would allow for a more like an even playing field, so to speak, because you know that you're implementing interventions that are the same across the board. So if you're looking for neuroprotection for our micro preemies, well, we know that they have all gone through the same essential golden hour admission. They've all been treated this way in the delivery room. Of course, there'll be outliers for ones that are transferred from all, you know, different centers, but for the most part, you know, all the care should be standardized, but, you know, let's try to take advantage of what's already in place, and then see if, you know, we can go there. So that would ultimately improve clinical trials and give us more of a, you know, straight, not a lot of variability with our outcomes and devices too. I mean, I showed you that picture. I mean, if we can try to standardize our devices, I implemented an IVH risk reduction QI project on our unit, and some of the devices were just not useful for what we have. You know, positioning agents aren't radiopaque, so you can't take an X-ray through it. So you have to pick up the baby, so that increases stim, and you know, so those are the things I think that we really need to focus on moving forward.
So from the parent perspective, and I know we talked a little bit about the barriers with families of opting into trials, the consent form issue. These are things that I'm hearing over and over again. From our perspective, and trauma-informed care, I think is a big buzzword, right? Your buzz phrase right now. But I think, really truly understanding, especially for our families, they are in the midst of trauma. A lot of the distrust in medicine might also be from their birth experience. And when you're in that fight or flight mode, having that psychological understanding of where families are. And I say this all the time, people walk through those doors with different baggage. Some it's a pocketbook, some it's their entire apartment. And really trying to figure out, how best to tailor that. And again, like for us, the plain language is so important, and having translation available, like already vetted through families, is very, very important. And using what we know from those lived experiences as you're building from the beginning, to make sure that whether that's, we're looking at a multi-sensory experience of consent, and you're bringing in, like I would have loved to have heard, again, we were out of the trial period, but, hearing from those parents that opted in originally to those cooling trials, from an outside, the medical perspective, it sounds like science fiction. Like I remember getting consented, giving consent for Max for cooling and transferring him. And, our neonatologist said, this is the available therapeutic. It sounds crazy. We know that it is shown to work with a lot of babies. And this is kind of the overview of what would happen with it. What do you think? Yeah. And, it was an easy decision for us because, and he's like, and it has to be done quickly. So we hate to rush you, but you need to figure this out quickly, know? And of course we're like, oh yeah, that makes sense. And how it was presented to us was in a very, short, succinct, effective way. And so I think just being mindful of that as, as these things continue, and just, again, the elephant in the room of the disinformation that is out there, and just not shying away from finding ways to best communicate with families.
So standardized and clear communication.
I was just thinking when you're talking, how frustrating it has to be. If it's frustrating for me as a nurse, how frustrating it must be for family that there is no standardization. So, you're here 1 week, we do it this way. The next week we do even the variability and standard of care amongst nurses. So I just think that must be very, very, very frustrating. So if we can standardize things and limit this variability, I think it'd be a lot easier for us to explain what we want to do, explain the trial to the parents, and that they can understand and not be so frustrated.
And I think the work that's been done in family-centered care standardization as well can very much apply to clinical trials.
And I think the key discussion too is around what to standardize. What are the key things to standardize? We can't standardize everything, right? And within that's why research networks, have been created because they have, agreed clinical protocols or standards. And, Sunny is Neonatal Research Network to do similar things and provide clinical and try to hold to. But I would say devil's advocate side is try to do it too much and standardize too much becomes not a pragmatic trial, right?
Right.
Implementation side of things are, can those out rigor those.
Right. I think we have time for maybe one more comment.
So I think there's a great example of the utility of standardizing care, and that is with the 22 weekers now. People are starting to resuscitate those babies in places that have really standardized the protocol, from the golden hour to, fluid and electrolytes to whatever, have really great results. Iowa is a great example. I think they have 60% survival and really good outcomes for 22 weekers. And most places it's like 15% survival. And so that just shows if
Yeah.
there's consistency of care, you do better. And the other point I would make is that, you can consent to a trial or not consent to the doctor who's on that day and whatever they decide to, give some medicine or therapeutic that isn't in a trial. So there's a danger in that as well.
There absolutely is. I have a dream of all the babies being enrolled in a trial. I'll hand the last comment or question to Kelly.
I just want to add one quick comment. Clearly, I talked too much yesterday, but as a neonatologist in a NICU that is enrolling babies in a lot of trials of a variety of scope, I would say that clinical research in a NICU is one of the greatest ways to standardize care. And the thing that I think helps my unit standardize care more than anything else is participating in research. And that standardizes our care for babies enrolled in the research, and that standardizes our care for the babies not enrolled in research. And I think that is one of the great quality improvement advantages of NICUs that are participating in lots of research.
Thank you so much. And I really wanted to thank the speakers, the entire panel, everyone who participated. It sounds like there could be some projects in here that Inc. might be able to take on to improve and help accelerate the field. Thank you.
So I wanted to start this Inc. Data Pioneer Awards ceremony. This is something that we started from last year. The idea behind this ceremony was to honor our collaborators and colleagues who have been who have been very graciously contributing data to our data integration efforts, which, as you know, for the past couple of years, this has been one of the biggest projects that we've ongoing at International Neonatal Consortium. And as a result, we have started this tradition of honoring our collaborators who have been very graciously contributing data to the Inc.'s real world database. So I wanted to take a bit of your time to actually make that happen today. But, before we do that, there was one more, category that I really wanted to honor today because, all of our colleagues at C-Path International Neonatal Consortium who have been helping us day and night, volunteering their time outside of their day jobs, doing all of those small and big activities, selecting the hotel, working with the AV people to make sure everything goes perfect, working with travel arrangements, reservations, and talking to meeting participants, please send us their slides and basically pestering you guys to make this thing happen, right? Setting up the meeting room, breakout rooms, Zoom meetings, slides, preparatory sessions, countless other small and big things that were really needed that happened on the back end. And they were needed to pull this type of a meeting, especially given the fact that for the past couple of years, we had become a little rusty because of COVID-induced isolation. Zoom meetings were always so convenient to conduct. So we kind of got into that habit of doing virtual meetings. We really wanted to break out of that habit and conduct this in-person meeting where we could actually meet our Inc. members in person. And personally, for me, as a fact, the only person who was fortunate to meet within the past 2 years was John Davis. And that's only once where I actually kind of self-invited myself to see him in person in Boston when Klaus was visiting there. But again, the point is we really want to do this in person. And I want to thank my C-Path colleagues, C-Path team for all of their help and efforts, time, and patience and persistence to make this happen. Can I have all of our So could I ask my C-Path team to stand up, please, so I can Um, I would like to thank my colleague Christine Barry, who has been two project managers and a project coordinator all in one package. I'm not joking. It's serious. She has taken up everything that we do at INC, whether older projects, real world data initiative, preparing for this meeting. I told her, this is your meeting. You do it the way you want to do it, and she did it with like remarkable, I guess, efficiency. And again, thank you, Christine. In addition to Christine, all of our C-Path pediatric team, Christine Miller, Huang, Grace, Bree, Colleen, Olivia, Gina, they worked in trenches, stood right behind by us, assuring us that they have all of the help that we need, working us late hours, early mornings, getting up at 5 a.m., making things happen. I could not do it myself. This is basically a teamwork. So thank you, all of you. In addition to this, I would like to thank my colleague Klaus and Dan, but still, for your intellectual and organizational support to make this happen. Obviously, Inc. leadership team, all of our leadership team members, John, Mark, Tom, our parent members, our nursing members, Bakako, all of us, right? So those were the people who actually encouraged us and basically stood by us as we spent a lot of time to make this meeting happen. So thank you so much. I greatly appreciate all of your help and your support and your collaboration with International Neonatal Consortium. So thank you again. Okay, so now I wanted to, yes, how could I miss Huang? I'm sorry, Huang, because I didn't see you. That doesn't mean that you are not a part of IMC's pediatric team. You are already, I already consider you as a honorary pediatric director who has been helping with all of the activities that go on behind the scenes. So definitely could not do it without you. Sorry, not totally non-intentional. Okay, so I wanted to get on with the awards ceremony honoring our colleagues and collaborators who have been very gracious with contributing data to our real world data initiative. And I'm just going to get to them one by one. Firstly, I would like to invite my mentor colleague, INC co-director, Dr. John Davis from Tufts Children's Hospital. Oh, we absolutely need to do that, John. Here you go. Come on in. Here is. And may I
Thank you very much. Sure. Thank you. This is a great honor from this organization. We've come so far, so fast, and yet for most of you, it's never fast enough. But we're on the journey together, and I really like that quote. That to go farther, we can do it together. So I'm really honored to receive this on behalf of Tufts and look forward to collaborating with all of you. Thank you so much.
And I would say that John Davis's support for our real world data initiatives, was really instrumental. It was, I believe, Klaus, one of the fastest data contributions that we've ever received for any of the ever at C-Path, right? So absolutely. The next one I would like to ask our colleague Mike Padula from Children's Hospital of Philadelphia. Thank you. We're honored.
Thanks, everyone. It's been great to be a part of this, and I'm really looking forward to sort of digging into all this data there and figuring out how to make the best use of it. So thanks for everyone, and thanks to my team at CHOP that did the heavy lifting to get the data.
Absolutely. That was, the data set from CHOP has been one of our highest quality data sets, electronic health record that we've ever received. So thank you. Thank you, Mike. Next one, Laura Fabry from Chiesi. Thank you so much, Laura.
And just a comment. On behalf of Chiesi, of course, I am emotional today because it's different to get this prize in face to face at last after virtually discussion last year and awarding last year. So I am appreciating much. And I want also to comment a little bit on our collaboration started some years ago. From last year, we grouped together many data and we created for the first time a new data sources for future application in real world evidence. So we have appreciated so much this activity together. And one of my dream, honestly, working in neonatology since long would be to achieve from these data sources a step forward in understanding bronchopulmonary dysplasia and achieving a good endpoint for future trials. Because I want also to tell you, and then I finish, in connection with what was discussed in panel discussion earlier, I want also to give you an additional point from pharma companies' perspectives. You are realizing more and more that after phase 2, many programs fails. And one of the reasons is certainly that, of course, the first reason that we do not have preliminary good results in phase 2. Of course, safety or efficacy, not intriguing results. But one of the elements that take a company to decide to stop a program is a no clear identification of endpoints, because until phase 2, we have proof of concept, we have exploratory endpoints. But when we enter in phase 3, we have to select one endpoint to have then in labeling, and we should have clarity. So this is clarity and also timelines. So for this reason, we need you, we need the academic people, Nikki Robertson, and also the others. We have already worked together. You need us for formulation, for money, for budget, but we need you to support endpoints and to give our colleagues in the company elements to trust a program. We have an endpoint, we have timelines, we have feasibility of the program. Otherwise, the programs are stopped in phase 2. This is one of the reasons. So academics need pharmas, but pharmas need academics in this discussion. So, and INC is an example of working all together. So we are honored for this.
Thank you. Next one is for our colleagues from NICHD. I would, we have been working with NICHD, getting data from their DASH database, which is, John, as you know, is something that Klaus, you also know, very first time ever that we got data from NIH and NICHD for our real world database. And our NICHD colleagues have been very gracious, very helpful, and expeditiously approving our requests to get the data. And I would like to honor NICHD for their collaboration.
It is a pleasure to working with this phenomenal group, and guys, if you need more data, come over.
Oh,
Keep it up, keep it up.
So we always need more data, and we definitely are going to take up on your offer. Thank you, Antonello. Okay, so we also wanted to honor Nina Modi from Imperial College London, and she runs the UK's National Neonatal Research Database. As a result of Mark and Nina Modi's help actually getting this data, we were able to get the largest data contribution ever in C-Path history. I believe before we got NNRD data, C-Path had something like 200,000 patients in their entire all of the other programs, and that just one data contribution instantly more than doubled our the number of patients that we have in our database. So it was something close to 280,000 patients that we got from NNRD, and we could not be more thankful for all of the help that Nina Modi provided and made this collaboration happen, especially given the fact that this is also the first time ever for C-Path's history that we got a very high quality data from UK as well. Unfortunately, Dr. Modi was unable to be present with us today because she had some other conflicts, but she sent us this video message that I'm going to play. I hope it works.
Hello everyone, my name is Nina Modi. I'm a Professor of Neonatal Medicine at Imperial College London and a long-standing member of the International Neonatal Consortium. I am so very sorry not to be able to be with you in person today. I had intended to be with you, but unfortunately I have had to be in Singapore at exactly the same time. So unfortunately it did not be possible. It's, however, a great honour and privilege to have been given this award, and I accept it gratefully on behalf of all of the UK neonatologists who contribute to the National Neonatal Research Data. Every single one of the NHS neonatal units in England, Wales and Scotland is part of the National Neonatal Research Database, and it's because of their collaborative spirit that we have been able to support this very, very exciting venture that is being delivered by colleagues from C-Path and the International Neonatal Consortium. I hope that the data that we've contributed will really make a difference to trying to bring to our neonatal units new treatments for the diseases and the conditions that afflict all of our babies all around the world. I wish you every success. Have a wonderful meeting. I'm so sorry not to be able to network with you in person, but look forward to catching up with many of you somewhere in the world in the next few months. My very, very best wishes for a successful meeting. And once again, thank you very much for the honour you do in granting this support. Thank you.
Thank you. Again, thank you to Nina for making this happen. Then in another series of firsts for C-Path and for INC and for C-Path as a whole, as a result of our very excellent collaboration with our colleagues from Japan, we were able to get a series of electronic health record data from our Japanese colleagues as well. And we have, for 3 of our Japanese collaborators. The first one is Dr. Kenricho Hosoi from Kyorin University. Unfortunately, he was unable to be present here, but I think he has sent us a video message.
Hi, I'm Kenricho Hosoi from Kyorin University and ICU. I'm sorry that I couldn't attend the local meeting. We are very honored to receive such a wonderful award. We would like to thank Dr. Hidefumi Nakamura, Dr. Satoshi Kusuda, and the many babies who provided data. I hope that we will use the data I have compiled to archive great results. Thank you very much.
Thank you. So one of the very important aspects of the real-world database is geographical diversity. We've been lucky to work with our collaborators from across the globe to get data from a variety of different geographical regions. Another one of our Japanese colleagues, Dr. Hiroko Iwami from Osaka City General Hospital, she worked very hard, in fact, personally working for many months to translate herself the data from Japanese to English language so that she can actually, transfer the data to us. We could not be more thankful to Dr. Iwami for her help in getting the data to electronic health record data to C-Path's INC's data integration efforts. Unfortunately, Dr. Iwami was also unable to be present in person, but she sends us this very grateful message. In fact, we are more grateful to our Japanese colleagues to make this happen. And basically, this is what she said. She's honored to accept this wonderful award. I wish I could be here with you, all of you in person. And, because of the time difference, it's always been difficult for our Japanese colleagues to participate. But I know this for a fact that so many times they have been here with us attending meetings when it is midnight their time. I personally would not do that. Okay. But, this is amazing. The amount of dedication, amount of persistence that all of our Japanese colleagues have. And working with INC at odd hours, and then working overtime to make this data contribution happen. So thank you again to Dr. Iwami for all of her work in contributing data. Finally, Dr. Tomohiko Nakamura from Nagano Children's Hospital, he also worked very hard to provide us with their electronic health record data from Nagano Children's Hospital. And he was also unable to be present in person today, but we wanted to honor him for all of his efforts for contributing data to INC's data integration efforts. Finally, this, is something that we always wanted to do. Everything we do at C-Path and at INC is for the benefit of patients. In INC's case, also parents, because parent members have been integral to every single thing that we do. It is basically our ultimate goal is to benefit and advance drug development for our basically constituency, that is neonates. We wanted to take a little bit of a time to honor all of our parent members who have been steadfastly working with us at INC, either being part of leadership team or being a part of various work groups at INC, spending countless hours volunteering with us, and in some ways trying to guilt us, to do the right thing, because that's exactly what we are here for. So to work for parents and their babies. So I wanted to take a couple of minutes to honor all of our parent members. And we are lucky to have a few of parent members attending this meeting in person today. I would like to ask Betsy Pylon, if she's here, Dabdai Senza, Katie, I apologize because I'm going to be very bad at pronouncing the name. Yes, Jan Daigle and Kiori could not be here with us in person, but we would like to virtually honor them. But we do have a special Friends of International Neonatal Consortium award for all of our parent members, and I would like for them to come on the stage and please accept this award. We would be honored. Thank you, Deb. Thank you, Betsy. Thank you, Katie. Of course.
Thank you. This is beautiful.
Yes.
I'm a little emotional. Thank you so much. Oh, how sweet. Okay, somebody will have to grab that. Otherwise, I'm going to drop this. Thank you so much for all you do. If I hadn't said it before yesterday, you really are the backbone of what makes this stuff happen. We're just there to lift you up and guilt you a little bit, but that's okay. It's important and we really appreciate it. It's nice to see things moving. And I want to thank the Inc. folks for all they do to put on these events, put on every meeting that we attend. They really do lift us up to do the right thing. Thank you. That's it. Yeah.
To echo Deb, I also want to thank you for the inclusion and attention to representation of more than just prematurity. Our families often feel very left out of conversations of, I'm happy to thrilled that there was a whole session about neonatal this morning, but it really means so much to our community to be validated in their stories and experiences as an equal part of the NICU landscape. So thank you.
Come on, say hello. It's okay. They don't bite. All right.
Thank you so much. I do appreciate on behalf of NICU parents, those that are in the NICU and those that have graduated from the NICU or have been given an opportunity to help further research. Thank you.
I would also like to say a word for Jennifer Deagle and for Corey. Thank you so much for everything you do for all of us parents. And Betsy's correct. We need to do a lot more work around HIE and other conditions. It's really important. Thank you.
Thank you, Deb. Okay, so the next upcoming session that we have is providing you with some previews and updates on our real world data integration activities, specifically the real world data and analytics platform. We call it as RWDAP. And we have a couple of our colleagues from C-Path who are going to be providing us with updates on the RWDAP platform, how it works. And we have Will Roddy, who's the data engineer at C-Path Data Collaboration Center. And I'm going to invite Will to come to the stage and provide us with the updates.
Thanks, Kamal. So what I'll be talking a little bit about today is some of the infrastructure that we've built around the real world data integration process. And I think it's kind of helpful to contextualize what the problem is that we're trying to solve. And this diagram that I have here, the goal is to represent that we have multiple healthcare systems that are delivering data to us. In the center, we have our real world DAP environment. This is our integrated database where we have curated that data to a single standard. That includes data that is a reflection of these patients' experience. So these babies that are born in the NICU, there are data that are derived from that experience. So there's things like prescriptions and medications. There's things like lab measurements and diagnoses that are made. But that's only one aspect of the story. And so building these high quality data are a way to help build an infrastructure to understand sort of what is that patient experience and begin to derive some insights from that data. One thing that is hard to illustrate, though, is what does that infrastructure really look like? And so in the center, I have a kind of an image of a database, but there's also a number of servers and kind of cloud architecture we build. And so one thing I'm going to hopefully try to illustrate today is what some of that data curation can actually look like once we kind of get to an endpoint by looking at a web user interface that helps illustrate some of the value of these data. One thing that I think Klaus talked a little bit about yesterday that's really impressive is the number of patients that we have from the Inc. Real World Data Project. So this is a graph of the data that we have over time from all of C-Path. And you can see that in the last year and a half here, the amount of data that we have from Inc. really exploded. And it's really quite impressive. We've eclipsed or are on track to eclipse the total data that we have from all other C-Path programs. And this just really exemplifies the value of this resource, but also some of the challenges that come along with being able to actually extract value from it and turn this into evidence. So a little bit about what I'm going to show today. This is a tool called Odyssey Atlas. It's a free open source software tool. It's developed by the Odyssey community specific for OMOP data. You don't really need to know too many of the details about that, but the idea is that this tool can help us explore summary statistics, define and evaluate cohorts of patients, and we'll be also able to use this to actually design and execute analyses against the data themselves. And it's all built within the infrastructure of being able to facilitate collaboration around these things. Just kind of level set expectations on what's up next, because demoing a website live is notoriously difficult. If I were to do it, the Wi-Fi would go out. And so instead, I have some screenshots. And I'm going to try to give you guys an idea of what it would feel like if we were to walk through this together. I'm going to highlight some features that we're currently using internally. And we're using this tool right now to help do some quality control and understand what data can continue to be curated. And so within the kind of platform right now, I have two data sets that are a work in progress still. So they're not finalized. I don't want you guys to think of this so much as a science talk where these numbers that I'm showing are official, but instead understand that these are numbers that are moving a little bit as we continue to curate the data. And it helps us understand where is the value possibly at. So kind of at the landing page, when you first log in, there's a number of menus on the left-hand side. The landing page includes some links to documentation and some quick get started links. This is all behind security firewalls, and there's authentication. And right now we've limited this to just C-Path internal users, but we're evaluating what possibilities may be there to help expand the access of this. The first tab that I want to call out is the data sources tab. So on this tab, there are these automatically generated statistics, and these are going to be present for every data source that we have. Right now, we have these for each of them individually, but we could also generate it for all data sources integrated. And if you hover over like this donut graph, it will show you the number of patients and the percentage of that population. And scrolling down on the page gives you a little bit more insights that actually begin to answer some of the important questions we have about what is the value of these data. So the cumulative observation shows over time, how many patients do we have at that point, which conveniently for this group is also how old they are. So because of the nature of neonates being seen very close to when they're born, we're able to see that there's about 75% at a year and a half of observations. There's a number of different statistics that are generated by sort of clinical event domain. These clinical event domains are part of the OMOP model that we utilize. And I want to call out a couple of them, but the details of these are going to look similar across all of them, even though I'm only going to show a few. So the person report gives us some of the demographic information here. Again, you can scroll over them. It gives you some interactive views. You can also export these as PNGs directly from your web browser. There's a data density report, which is really useful for helping us understand what the data quality is over time. So this gives us the total rows or the number of records that we have in any clinical event domain. And this is useful from the perspective that we can say, well, how many measurements are there over time? But also looking at this, we can say, well, there aren't a whole lot of observations in this data set. And why is that? And is that potentially a cause of error, or is this something we can go back and curate further as we work through the data? It's sort of similar. This is also interactive, where if you hover over it, it will show you the records per person, for example, at a point in time for that line that you're on top of. And there's also these box and whisker plots that show you the concepts per person. So the example here being the observation doesn't quite follow the rest of the clinical event domains. And so we would want to evaluate what is the reason for that. There could be very good reasons that that's still fine from a data quality perspective, but it also could be a concern. And then some of these extreme outliers, we might want to look at those specific patients to understand, are there maybe duplications in their records when the data was extracted from the source? There's also details on the observation period, which due to sort of the young nature of these populations don't have particularly interesting distributions at this level, but we are able to get a better visualization of how much data is there. So for example, there's this histogram that shows over time, how many patients do we have in any of, how many patients do we have by the number of years that they were observed. This is an example where we actually dive into some of the clinical events. So this are measurements. Measurements are things like vital signs, anthropometrics, lab values. And so in this data set, some of the most common or most prevalent measures were body weight and body height. And we have the number of persons with those measurements and their prevalence in the population. There's also the records per person. And this is a really useful metric to understand what's sort of the data density at a top level. So here we see that we have body weight and height, and that weight is measured much more frequently than height. We would expect that. That's consistent with the fact that your weight changes more often than your height would. And systolic blood pressure and diastolic blood pressure similarly are measured much more frequently than what we see for body weight. It's also possible to filter by string, so you can search in a text box. So here I've searched for eosinophils. You can, if you were to click on one of these rows, generate a drill down report where we have details on the prevalence of that given measure. And we actually are able to create breakdowns by the units that are present within the data and the distribution of those data by unit. So we see that we have a little bit more than 50% of these are reported in percent units. We have the value distributions as box and whisker plots for any of the units that are present, which help us sort of understand what variation there may be. There's also these values relative to normal range, which is a challenge in this population. And Carol and Nick are going to talk about that more in a minute here. The second tab that I want to call out is the search tab. So on this tab, you can search for clinical concepts or terms. So just coming in, you can search a term like birth weight, but this can be any term that you're interested in. So it's disease agnostic in that regard. And so searching birth weight, the first thing it shows us is a birth weight finding. So this is a kind of condition or an observation about a patient. The other thing that I'll call out just at this level is there's a column for RC, which is record count and descendant record count. So at first glance, you understand within this data set, where are there actually records that are relevant to these clinical terms that you've searched for? And if we click on birth weight finding, the descendant record count is related to this idea that concepts can be organized into a hierarchy. So birth weight finding, for example, you can have more specific kind of children concepts, which are a birth weight of 2 to 2.5 kilograms. And this becomes really relevant for drugs where you can think about, say, you have ibuprofen, but you can also have more specific terms like you have ibuprofen 500 milligrams. And so this breakdown gives you a sense of where is that distribution of data within the hierarchy and where can we begin to find commonalities of the data. So it may not be possible to find common data all the way down at a branded drug name, but potentially we could roll things up into an ingredient or to a therapeutic class and be able to do some analyses. And this allows for us to, at a very quick level, see where do those data exist within our database. And then sort of similarly, there's a record count tab, which will show you across each of our data sets, where are those records at that level of the concept or at any of those descendants. So the descendant concepts is useful because that gives us an idea of there's more granular detail that we could go to, or if we've already gone too granular. One thing that's really kind of a nice feature of this is that you can collect these terms into what's called concept sets. So you can think of this as sort of a shopping cart, where maybe you come along and you have a number of respiratory medications that you're interested in, and you could collect those into one concept set, one unit. And then that's something that could be used for collaboration. So for example, I've started building respiratory medications. And if we click into this, I've begun to add some respiratory medications that I understand are of interest. And I'm sure there's many more that we would want to add. So these are some surfactants and sildenafil. And you can use these for a number of different kind of features. One is it's just a nice way to kind of collect them all in one place and know these are all the possible source terms that we're looking at within the data set. But the other thing that we can use then is for building cohorts and beginning to actually develop analytical subsets of the data. So a little bit of terminology around patient cohorts when I'm talking about that. I'm talking about a set of patients who satisfy at least one or more inclusion criteria for a duration of time. And so this is used to group patients with similar characteristics in analyses. And maybe patients who all had a similar event or have similar outcomes or have had some similar feature with them. And it should define specific logic for that and criteria for the inclusion and exclusion of those patients when they enter and exit a cohort. And we can define these two different ways. One of those is with rule-based definitions, where we use subject matter experts and domain expertise to generate a set of bounded rules around these things. There's also probabilistic definitions that use machine learning. For today, I'm going to show us an example of what a rule-based definition may look like. Again, kind of with the underlying understanding here that this is just an example. And yesterday during the BPD group, we had a lot of really good discussion about what we may want to use to define these populations. But this is one possible interpretation of how we can begin to sort of conceptualize these problems. So when we start building a cohort, we have to ask ourselves a number of questions. What clinical events are meaningful? What representation of the patient's experience do we want to make sure that we're including in this? What clinical events define the cohort entry? What are the inclusion criteria that we apply to those events? And what defines the cohort exit? So this paper, the CPIP, which again, to kind of call back here, that it's kind of funny to talk about other people's work when I know they're in the room, and a number of people here know this paper much better than I do, but I understand that it's relevant. So this has a really nice sort of exposure progression where there's enrollment down to long-term endpoints, and this really follows that model of what a cohort would look like. And the paper very nicely has this checklist, which for an informatics guy who doesn't necessarily know too well what's happening in the NICU is great because I can translate this into data talk. And so here we have the list of meaningful clinical events. We have the time of cohort entry, which the paper tells us is 36 weeks PMA or discharge, and that we should be looking for these clinical events in the last 7 days. And then there's the inclusion criteria, like FiO2 has to be greater than 21%. So there's a number of very bound metrics that we can apply to a data set, but we still need to go through that process of actually translating all of this into concepts. So for example, medications for a respiratory sign or disease is a fairly broad kind of component, but we can take that and translate that into the ingredients that would be found in surfactant drugs. And so that's where we begin to have this balance between building a concept set and building a cohort. And so within our example cohort, I created this sort of visualization that brings together some of these different components, a little bit of what the paper had, a little bit of what I've talked about, where we have kind of at birth, the patient and the patient's experience. There's going to be a number of clinical events that are happening. There may be some antenatal risk factors ahead of that. There's going to be the entry event, which is 36 weeks PMA or discharge. And then there's a negative look back of 7 days where we ask, have any of these inclusion criteria occurred? And then there's going to be possible outcomes. So the cohort that I'm going to show you guys what this would look like is the process of actually defining that cohort for the entry event, what the checklist has, not necessarily anything with outcomes. So on the cohort definitions tab, we can come back and we can say that the entry events are that we need a postmenstrual age equal to 36, or we want to visit occurrence of inpatient visits for the first time that needs to end at some point. And so we have either of these two entry events, and we can apply rules around what those inclusion criteria are. So for example, here I have the FiO2 is greater than 21% that also has the logic around maintaining an SpO2. And we also have some rules around, or there could be a procedure occurrence or a device exposure or a drug exposure, where we begin to capture some of these clinical events as the data is interpreting them. And one thing that's really cool about Atlas is it also includes a graphical view of this cohort, which is nice for kind of a gut check evaluation. So when you're done, this is generated automatically. And this matches our sort of mental model, hopefully, that we have at day 0, which is this entry event. And then if you look back 7 days, we're looking for any of these occurrences. And then right in Atlas, you can actually generate this against any number of data sets that are currently available. So just clicking the generate button, we'll find the number of patients who meet the entry event criteria, and then the number of patients who continue to meet any of the inclusion rules. And we get a stratification of those data. You can also create similar reports like what the data source summary was for that cohort specifically, and understand what are other components of interest here. And so this is an example of procedures that describes what procedures are prevalent in this cohort. You could also do this for conditions or medications, where you begin to get a more nuanced breakdown of what are the details of those data that are associated specifically with this group of patients we're interested in. You can also do characterizations of the data. So characterizing the data includes things like defining the cohort and then defining a number of features like any measurement and defining what those measurements over time were. You can also define these features ad hoc in the system directly through a graphical user interface, or we can define more nuanced ones on the back end directly through coding. So these are fairly customizable. And to kind of give you a sense of what these would look like, we can look at other covariates that are present within this population or between two populations. So here I'm comparing a low birth weight cohort with a very low birth weight cohort. Not necessarily for any expectation that they would be different, but to see that in this case, anything that doesn't fall sort of on this line, the dotted line in the middle, there's some difference between those two populations. And so this could become really interesting when we're looking at different outcome cohorts. And it also generates a number of graphs that help us explore the data at a top level. There's also incident rates. So incidence rates take a target cohort and some outcome cohorts and calculate what the incidence of that outcome for a target is. So the idea of the target cohort being this is a patient population of interest, they have some shared event, and then we want to see what happens to them later. So we can take a look at how these patients fall into any of those outcomes. And you can also define stratification. And so you can build right into the system possible, for example, if you wanted to stratify by very low birth weight versus low birth weight, how do these incidence rates compare between those populations? And so here's an example of that BPD checklist versus pulmonary circulation disorders, where we've stratified over a number of factors. And you get the number of cases and then the proportion per 1,000 persons that meet these criteria. And so we get sort of an epidemiological understanding of what is the presence in this data set. And this is really useful for understanding how two data sets may be different or similar. And also, do we have enough volume of data to start answering some of the analytical questions that we have? So last thing I'm going to call out is that there are a couple other features I didn't quite have time to get to today. These are things like cohort pathways, which are going to explore how patients move between different cohorts. So for example, if a patient starts in cohort A, then do they go to cohort B or cohort C first? There's profiles where you can actually look at one patient at a time and see what is an individual patient's pathway and what are all of the events that apply to any one patient, get a sense of at a more drill down level, how does this affect the individual? And then there are estimation and prediction modules. And so these modules help us actually define the statistical analyses. And there's a number of parameters that you can set right within the user interface. And then it generates a downloadable R package for you to run the statistics directly against your data set. So hopefully what I want to illustrate here is that this is just the beginning. It's one way that we can begin to interrogate the data and understand it, but it builds a nice user interface around what those possibilities could be. And this is one way that we can expose it, but underlying this, we have a number of infrastructure tools that really help push us even further. And so this is the sort of the start. And we have these components. Hopefully this can help the working groups build questions that we can derive answers to. Thank you.
Will's talk was more focused on how the data when focused towards BPD specific uses might look like and how the graphical user interface of our data platform looks like. Next up, we have a presentation by Carol Elgart, who's one of our collaborators and faculty at the University of Leuven, Belgium. He is going to talk about the second part of our real world data analytics platform project, that is the neonatal lab values. Carol, do you want to?
Yeah, the big one. Thank you for your kind invitation. I was asked to present in about 10 minutes the results we collected on a structured literature review. And actually, I was also asked to illustrate a little bit, why are we doing this? I mean, how could it be that we don't have data yet on what's a normal value for sodium or potassium or whatsoever? And the second question that I was asked to answer is, why not the easy way? Because people must have published already on that. So why don't we simply pick the low-hanging fruit? And unfortunately, it turned out not to be the case. So when working through this project, we realized that perhaps there are still some deficiencies to really make it into an impactful outcome. So we all should realize that at present, there are no generally accepted age-appropriate reference values at present for at least lab values in newborns. This matters not only for drug development, but that's the reason that we are here today, but also for clinical care, for safety aspects, for whatsoever. So it's broader than just drug development, but the reason we are here is with this focus on drug development. And as we all know, Inc. has engaged to define actionable reference values of what we call commonly used lab values in newborns. And to further explore on the why, and again, it could also relate to efficacy. We have heard on the peanut and other studies, but it could also relate to safety. And how do you recognize an adverse event? How do you compare an adverse event? I mean, imagine that somebody wants to use ibuprofen, another one wants to use indomethacin, and both believe that is the best choice. And we see some difference in kidney function, which is indeed the case. But how can we quantify these differences? Is it a little bit less and a little bit more? That's not the way you really want to work. So if you want to grade severity, you want to have absolute values and have to have reference values to compare to. And that's what we lack. I refer to the work which has been largely done by Thomas a few years ago within the Inc. initiative. And at that time, we had to decide, well, sorry, lab values, we will not touch on that because nobody knew what to do with it, right? And that's a little bit uncomfortable. Did that sound really scientific? Let's be honest on that. I mean, but at least I think we have learned by this approach that actually what we have to do in our profession is to convert what already exists in other populations and to make it applicable to the newborn. And that's a theme that I would like to come back to, because we have to realize that we have learned a lot from already existing systems and have adapted this to, let's say, the neonatal setting. And I will simply like to show you two data sets. And the first data set is on creatinine. And I'm sorry for those who work in millimole, but this is European, at least my Europe. So it's a milligram per deciliter. I'm sorry for those. But this is simply plotting data from my unit. 5,000 observations in a little bit more than 1,000 kids. And what happens in the first 6 weeks of their life, creatinine goes up and it goes down. That's physiology. There's nothing wrong with that. That's how a baby behaves. But I think that you don't need to be a great statistician to realize that the median trend value is not a really good predictor because you have extensive variability. And this variability is there from the day 0 onwards. It to a certain extent explained by clinical characteristics. That's one aspect, what's normal for a given individual. And the second aspect relates to the fact, what we can use to recognize kidney injury is AKI. And AKI is defined by an increase of 0.3 milligram per deciliter. Then you have a stage 1 or grade 1. Well, grade 1 is physiology for an extreme preterm. They all go up. So it depends. You can say all babies are respiratory insufficient and there are kidney insufficient. That's perhaps a way to look at it. But still, you want to make a difference between somebody who behaves physiologically and somebody who disbehaves on a physiological base, because it can be induced or related to drugs, either positive or negative. And as the former session was on asphyxia, I subsequently tried to apply this to a creatinine data set collected in 1,000 babies who undergone therapeutic hypothermia. And this is a little bit a complex approach, but we call it the Pottle approach, likely because Pottle is one of our faculty members. But he has gained quite some credibility throughout the world related to creatinine values in humans. And what he has done, what he suggested is just use ratios. So what I've plotted over here is the former slide, but only in term babies. Imagine that it was 0.8 milligram at delivery, and an asphyxiated baby, it would be 1.6, then the ratio would be 2. Yeah. So actually, if you would plot simply centiles over time, you would have different lines along the line of the 1. And what you can see, yes, babies who are going therapeutic hypothermia, they are on average born with poorer kidney function. Majority of kidney dysfunction disappears over the first 2-3 days of life, but it never becomes normal in the first week of life, right? And again, some babies, that's the way I try to explain to my students, some babies forgot to develop kidney dysfunction after asphyxia, while others had major kidney dysfunction. So again, you have a lot of variability. Part of this variability is driven by severity of asphyxia and whatsoever, but still, we need this type of data to better understand and to compare different interventions and whether or not they are protective for kidney or not. Same holds true for liver function, DILI. Quite interesting, and I was recently involved in a data safety monitoring board with DILI for an asphyxia-related drug. And it was known it was one of these repurposed drugs. It was known to cause drug-induced liver injury in children. So there was an ASIA of interest. And then the simple question was to us, poor neonatologists, what is a normal kidney enzyme, normal liver enzymes in newborns? I don't know. And in asphyxiated cases, it will be again different. And what you can see is, on the one hand, work that has been done by, amongst others, Mark Turner, who illustrates that postmenstrual age is one of the drivers. So if you look at normal liver values in a preterm compared to a term baby, it differs. And the other side of the slide shows you the asphyxiated cases in which degrading will affect normal values. So again, we need this type of data. And actually, we are currently building such a data set in about 2,000-3,000 cases and try to put it in the public domain to say, here, this is how these babies on average behave. So we need this type of data. So why not go for the low-hanging fruit, right? If we all publish data, why don't we simply harvest what is in the public domain? So this is what we intended, to create an overview on publication quality of lab values in clinical studies. And we first developed a questionnaire to screen on information completeness. We, in the pilot group with Mark and Heidrun and the chair of the session, we created a provisional version. We went to the Inc. real world data set to discuss this, and we came up with the questionnaire actually reflecting clinical characteristics, lab type of characteristics, outpatient, inpatients, assay type of use, and this type of stuff. And we subsequently did a structured search assisted by the colleagues of Bayer and ended up with 257 data that we had to extract. Unfortunately, we had to conclude that lab values for neonates are sparse, not systematic, and incomplete. Sometimes it's clear from the paper that there were pair data, but these data were not analyzed in this way, for instance. These type of basic things. Or you couldn't figure out which type of drugs these babies were exposed to, or you couldn't know what were the clinical characteristics of these babies. This obviously undermines any effort to compare treatment, safety monitoring, and clinical management, and to go for data extraction. Actually, there was a very limited number of data papers who said, yeah, you can reach out to me to get my to consider to share raw data. So this actually supports the real world data work, which is currently ongoing. And finally, there appears not yet to be a standard. And this is, I think, also an important message to consider today, not yet a standard for lab value reporting in newborns. It's quite different from how a clinician within the clinical context, that if you have an abnormal lab value, that you will have a report one way or the other to the clinician from, hey, this is a real high potassium level, or this is a real low glucose level, this works. But this is a little bit different. We don't have these standards. So how do we make progress? Do we have to reinvent the wheel again? I don't think so. Because what we have done with let's say, the adverse event score is use what has been used already in the literature and by other groups and by other, with focus on other populations, and try to convert it to how does a baby look like? This is what we have in common as Inc., right? So if you look in the literature, our problem is not so unique. There are also lab value type of journals or lab focused journals who say, well, we need much more information. What was the assay? We all know, like for creatinine, if you use Yafe or enzymatic, that you can have a different absolute value, right? And so there is on the one hand the call for more lab-related values, but the other hand, there should also be clear that the baby is not a small adult, and that we as an Inc. community should come up with a standard. What do we want to know? Age characteristics, weight characteristics, drug, whatsoever, things that we value. So my suggestion would be that we will further work on that and also develop such a standard for reporting literature, and then assist it, as use this with their own real world data, and also provide to the community to further assist in this ongoing project. And I think this is likely my last slide. So I had a result and also some work to do. And I would like to stress the fact that the job is not yet done. Thank you very much.
Carol, so Carol's talk was more about why do we need to generate these, I guess, reference ranges that are generalizable, and the rationale behind that. And Nick is actually going to talk about how we are actually going to do that, some ideas about some of the analytical methods, and what sorts of considerations we need to take care of in order to actually do the analysis on the data. And Nick Henschfeld is a quantitative medicine scientist within quantitative medicine at Critical Path Institute. So Nick, you're on.
Yeah, thanks. All right. So I'm going to give just a sneak peek into the directions that we're going. As Carol mentioned, there's a lot of work to do to understand how to take the data that we have in the Inc. database and use it to produce these reliable reference ranges that follow some sort of criteria that are standardized. So I'm going to give just a little bit of a background and then some very preliminary results from one of the data sets that actually has a lot of these measurement values for neonates. So just as a refresher reminder for those that need it on statistical methodologies for developing reference ranges, or how you would actually go about doing this, and to explain, why we can't do the typical statistical methodology that you would normally use if you were a laboratory publishing your reference ranges for your specific values. Typically, you would recruit a cohort of patients that you would satisfy some criteria as being normal patients. Test them, get the lab values for those, and then have a very nice statistical distribution that you would be able to extract your 95% reference range from. And obviously in neonates, that's a recruiting problem. It's almost impossible to define what it actually means for the patients to be normal in the first place. As Carol was mentioning, it's difficult to tell what is pathological versus physiology in these patients. And then it's just also difficult to recruit. So while it might be considered the most rigorous and reliable and follow certain guidelines, it's basically impossible to do. So we're stuck with using real world data, but that's why we're all here. And so really the only difference is that we now have a greater statistical job ahead of us in defining how to extract the data from these databases, and then the statistical methodologies used to develop the reference range from that. The distributions are going to be potentially skewed. They're going to be potentially mixture distributions where you have both normal, whatever that means in this population, and abnormal patients. And then there's also going to be the problem of time dependence and covariate dependence that we have to deal with. So we're currently looking at a database that has measurement values. We currently only have one EHR database that has about 12,000 patients. I'm currently using a training subset of that that's about 2,900 NICU patients to develop the statistical methodology. So again, this is all very much in the development phase. As I understand, there's 2 additional databases that we're getting soon that have some measurement values, but it's going to be a data challenge to extract those values. They're not mapped into the nice OMOP format. And as I understand that with the database that we currently are working with, all of the measurement values that we would like to target, which is a long list shown in this paragraph on the right from the INC grant application. As far as I understand, we have all of these lab values in our current database, and we should be able to develop some sort of reference range for most, if not all of these. But they all might present different challenges. So we'll have to do a process of prioritization to figure out which of these we want to target first. And like I mentioned, that there's some unique considerations in using real world data, namely that there's potentially reporting errors, potentially inconsistencies in reporting, a lot of the mapping problems and the data problems that we will mention at some point. And then we also have to deal with the time dependence and dependence on other covariates like gestational age. So one of the methodologies I've gone through the literature, the statistical literature on dealing with what are called indirect reference range studies or reference ranges from real world data. And the one that is sort of recognized as the state of the art is a methodology called Refine R. And it's an R package that you can use to apply to real world data. And it makes a very limited set of assumptions about the structure of the data. And it's able to extract a reference range or reference interval, even if your data is not perfect, as ours will definitely be. So it allows for skewed and multimodal distributions. It allows for a collection of so-called normal and non-normal patients in the population. And it uses a statistical transformation methodology to fit a unimodal distribution to the data. And they provide good evidence based on simulation to show that their methodology is extremely high performing and extracts the correct range in many cases. One of the cons of this methodology that I'm running into is it's not clear what the dependence is on the size of the data. I mean, we have a few thousand data points, but it's never clear. You know, in traditional methodologies, you would be able to calculate a population size that would be sufficient. the number that is typically used is 120 as a sufficient population size to do a direct reference range calculation. In this case, it's not clear, we seem to be getting reasonable performance with 2,900 patients, but it's not clear if we need many, many more than that to get the results that we want. So in a very preliminary example, I extracted bilirubin values for this cohort of 2,900 patients and use this refine-R methodology to generate a reference range. This is very preliminary results that are depending on a lot of further analysis to make sure that we've made the correct assumptions and extracted the correct values. But so far, it seems like we're able to pull out a reference range. And this is, by the way, for the values, the very first value for each of these patients, because like I said, and as Carol showed in his presentation, these values depend on time post-birth. And so this is just the very first value for each of these patients. And so in theory, we could generate one of these plots for over time and see how that varies. So the next steps beyond this are using outcomes potentially to split the population into different subgroups to see what the dependence is there, and then exploring dependence on other covariates like gestational age, and then looking at issues like cross-validation and robustness to see how robust these results are on various assumptions and modeling, and then looking at additional subjects when those come online. Thanks.
Thank you, Nick. So that was great. I mean, the purpose of this session was really to give you some flavor about what is actually happening on the back end by when we're starting to do the analysis to create those models for bronchopulmonary dysplasia, and what is happening in the back end when those reference ranges are being generated. Definitely wasn't supposed to be an exhaustive lesson on statistical methodologies or database construction or anything of that nature. It was just to give you kind of a high-level overview of what goes on. And because we wanted to be inclusive, we wanted to include our data scientists and quantitative medicine scientists in this conversation also. So because we were running about 15 minutes behind, I'm really especially thankful to Will and to Carol for making up the lost time that we lost earlier today. And we're actually right at 11:45 for the next session. It doesn't seem to be working. This one is regulatory updates in neonatology. The purpose of this one, because we've heard from a lot of our members in industry and academia. They wanted to hear directly from our regulatory colleagues from FDA, EMA, PMDA in terms of what actually happens behind the scenes. One of the things that they were really interested in was talking about the cluster calls initiative. So I'm going to ask Jerry Baer, Sarah Zaidi, and Ralph Bax and Hide Nakamura to be on the stage and provide us with some updates on regulatory affairs, so to speak.
Thank you, Kamaljeet. While folks are coming up, I assume everyone knows that Hide is not with PMDA, but he'll be representing PMDA. And I believe you have comments from Junko? Or, oh, come on. Thanks. So thanks to all of my colleagues. I'm just going to give a couple of very brief updates from the FDA, and then I'll turn it over to Dr. Zaidi to provide an overview of the pediatric cluster and how that works, and then over to Ralph and Hide. So from the FDA perspective, we have finalized, so a couple of general updates. First is guidance. We finalized the neonatal clinical pharmacology guidance, so that is out. There is a new pediatric growth measurement guidance, which includes information about measuring growth in neonates, which is out in draft. So please take a look and provide comments if you have them. We are certainly looking forward to the comments. The pediatric clinical pharmacology guidance, which I think originally came out in 2020, 12 or 14 is out in a new draft form. There were enough changes to the prior draft that they issued a new draft rather than finalizing that. And so I believe that's open until the end of the year for comments. And I would just say, keep your eyes and ears open. There will be more neonatology oriented guidance to come in 2023. So keep your eyes open for that. Another quick update is that with my colleague Ann Massaro and some of the other 5 neonatologists at FDA, we have been working and initiated the planning of a workshop in March of 2023 with the Duke Margolis Center. And that workshop is going to focus on the general issues of measuring clinical benefit. How do we know that neonates are getting better in our neonatal clinical trials? And so some of you will be hearing from us as we start to fill out the agenda and figure out what we want to discuss for that workshop in March of 2023. I believe it's March 23rd. If you wanted to save the date, it'll be an open workshop so anyone can attend. I also, the last update I have before I turn it over is that as of Monday, Dr. Ann Massaro, who you have all met now, will be taking over as the team leader for neonatology in the Office of Pediatric Therapeutics. I will remain at FDA, but in a different capacity in the Center for Drugs. So looking forward to turning it over to the next generation or something like that to carry the torch and continue working on behalf of neonates in FDA. And so with that, we'll introduce my colleague, Dr. Zaidi, who's a pediatric pulmonologist and a colleague in OPT.
Thank you so much, Jerry. I would really want to thank the organizers of INC and C-Path for inviting me to be here to speak. And if it wasn't for Jerry, I wouldn't have this job and I wouldn't be here to speak. So I really want to thank Jerry and acknowledge her for that. Also want to say that it's kind of strange to talk about the cluster, and while Susie and Ralph are here, I'm a newcomer on the scene and I feel a little bit of an upstart talking in front of them. So they have the institutional memory and hopefully they'll add their knowledge as we go along. I wanted to just get a show of hands. How many people know what the pediatric cluster is? Susie's got 2 hands up. Okay, it's a fair number. That's great. So hopefully at the end of the next 10, maybe 15 minutes, you'll know a little bit more about it. All right, I think I have my, let's see. It is, the big arrow. Okay, there we go. This is my disclaimer slide. You've probably seen this before, so I won't go through it. So the objectives of today's talk, we're going to kind of take a step back in time. For those of you who are history buffs, you'll enjoy this part. We'll talk about the history of pediatric legislation in the United States and the EU and describe similarities and differences in US and EU pediatric drug development. Of course, it comes out of the legislation. And we'll describe the history of the pediatric cluster, and I'll show you how, with the development of pediatric legislation, the pediatric cluster kind of went along lockstep, hand in hand. We'll review the processes and procedures of the pediatric cluster to sort of demystify or, just increase general knowledge of how we function. And we'll talk about how some of the outputs of the pediatric cluster since inception and how the cluster supports international regulatory collaboration in neonatal drug development, which is, I think, the topic that is more near and dear to everybody's heart here. So I think of some animations. I just want to give a little bit of a introduction to the layout of this slide. There's a couple of slides that are set up this way. The top half is in this light blue. You see the light blue box. That's for the United States portion of whatever that slide would be. The bottom is the green box, which represents the EU. And so that's a format that we have for the next upcoming slide. So the top of this, you see that in the United States, there's a whole bunch of boxes. In the EU, there's only 2 boxes. And what that basically shows you is that in the United States, legislation related to pediatric drug development started much earlier. from 1979, really, when product labels started including pediatric use section. And then the dates that we really refer to over and over again when we talk about pediatric legislation in the United States really starts in 2007, when the FDA Amendments Act reauthorized the Best Pharmaceuticals Act for Children Act and the Pediatric Research and Equity Act. In 2007, the Pediatric Review Committee, which we call the PERC in the United States, was developed or established. In 2012, both of these pediatric regulatory processes, the BPCA and PRIA, were made permanent by FDASIA, or the FDA Safety and Innovation Act. In 2017, a very important piece of legislation for our pediatric oncology colleagues was enacted. The FDA Reauthorization Act of 2017 included the RACE Act as well, and this allowed FDA to require studies in pediatric oncology. And in 2019, I won't talk more about oncology because it's not that relevant here, but going over to the EU side, you'll see that in 2006, the pediatric regulation was adopted, and in 2007, it had entry into force. And the Pediatric Committee on the European side of things was established, and that is popularly called the PDCO. So it's PERC in the United States, PDCO in Europe. That's where that stands. So let's talk about what the regulatory process is for new drug development, both for the US and the EU. And again, this slide on the top will show the US portion and the bottom, the EU portion, as regulated by the EMA. So in Europe, really, the discussion about the pediatric drug development starts at a much earlier phase. So in phase 1, you should have an agreed pediatric investigational plan, or PIP, or a waiver. You should have obtained that in the phase 1 part. Whereas in the United States, the first agreement between usually sponsors and the FDA, the initial pediatric study plan, takes place in phase 2. We do have another regulatory method by which we can talk about pediatric studies and talk to sponsors about pediatric drug development, and that's the written request. That usually comes up around phase 2 and phase 3, but you'll see that it can also come up in the post-marketing phase. And then once you have this IPSP, or initial pediatric study plan, that's agreed, or you have a written request. Oh, it said, did it suddenly go off? No? Okay. You can propose modifications, because as you know, things change, information changes, data changes, what we know about something may change. And in the US space, these modifications can be proposed by the FDA or by the sponsor. This is a little bit different than how it works in Europe, where modifications to the PIP must be proposed by the sponsor. The EMA does not have a mechanism by which they can reapproach the sponsor and say, hey, we propose that you modify this pediatric investigational plan in the following ways. So after phase 3, you have your NDA or BLA, new drug or biologics licensing application submission in the US, and you have a marketing authorization application in the EU, and then you enter the post-marketing space. And in the US, in the post-marketing space, there are still interactions between FDA and regulated industry, where we could still be issuing written requests in that time frame. There is a pediatric review of safety that occurs. The PMRs, or post-marketing requirements that are related to PRIA, the Pediatric Research and Equity Act, they can be issued. And also pediatric studies, even those that may have been agreed at an earlier timeline, they're typically conducted in the post-marketing space for all of the pathways, the PRIA, BPCA, written request IPSP pathways. So let's move on to the global priorities in drug development as stated in the ICH E11, or the International Council for Harmonization E11. This is a long paragraph, but usually I don't read out my paragraphs, but I feel like the screens are a little bit dispersed, and at least they were hard for me to read from. So I'm just going to read it out, if you can bear with me. Pediatric patients should be given medicines that have been appropriately evaluated for their use in those populations. Safe and effective pharmacotherapy in pediatric patients requires the timely development of information on the proper use of medicinal products in pediatric patients of various ages. Obtaining knowledge of the effects of medicinal products in pediatric patients is an important goal. However, this should be done without compromising the well-being of pediatric patients participating in clinical studies, which I think everybody in this room already knows. This responsibility is shared by companies, regulatory authorities, health professionals, and society as a whole. So that last sentence there with the phrase regulatory authorities, my colleagues and predecessors took that, and this is a reference to a published paper by Penkov and colleagues, which states that the FDA and EMA are committed to the ongoing harmonization of scientific issues and convergence of approaches through the work of the pediatric cluster, with a view toward a more global approach to the effective and efficient development of medicines for pediatric patients, including trying to save exposure to unnecessary or duplicative trials in pediatrics, and that includes neonatology. So to this end, there was conversations about establishing the pediatric cluster. And again, this slide is laid out the same way, with the US on the top and the EU on the bottom. On the US side, you can see that in 2003, FDA and EMA signed a confidentiality commitment that allowed them to exchange confidential information. In 2004, the first cluster was established for HemOnc, but we will fast forward a little bit to 2007. In 2007, and this is the same time, just as a reminder, that the pediatric review committees were being established both in the US and the EU, the first pediatric cluster meeting was held. So the pediatric cluster was established, and the first meeting was on July the 26th, which was even before the first PDCO meeting. So this slide just goes to show that the pediatric cluster has come up and it was sort of born at the same time as a perk in the PDCO, and we have a long history of working together ever since we started working on pediatric applications together. All right, this slide is just to break up the monotony of all those words that you've been probably looking at since the morning, and to display, really the agencies that at this time participate in the pediatric cluster. And all of these participants, highly intelligent, accomplished, dedicated, personable, and I don't know if it's politically incorrect to say beautiful ladies that participate. So we have Franca from EMA. She's been handling this since the EMA, has been in the pediatric cluster since 2007. For Japan, we have Junko Sato and Michio Sakiyama. From Health Canada, we have Agnes. Agnes asked me to say hello to everybody. She wasn't able to be here in person, but she says that she really enjoys these meetings and she hopes we have a very productive meeting. And for TGA, which is the Australian representative, we have Yeshwant Sinha representing. So what are the objectives of the pediatric cluster? These are we have a bunch of objectives. The first is to facilitate regular exchange of information between agencies that have confidentiality commitments. So not only does FDA need to have confidentiality commitments with everyone in the cluster, but they need to have it with each other so that we can discuss information in a safe space. We aim at a global pediatric development. We want to understand the scientific rationale behind our thinking, especially when differences exist. And one of the objectives of the pediatric cluster is to discuss post-marketing pediatric requirements. We also discuss general topics, and we'll talk a little bit more about that. And to inform participants of ongoing work that may be going on in different agencies where there may be scientific meetings or workshops, and to open the opportunity of actually attending those meetings. So the pediatric cluster was established in July 2007, and this information gives you a little bit of output of the cluster up till the point of June 2002, so this summer. This is the main forum for harmonization in the regulatory space for pediatric development and in the international context. And I don't want to get too fixated about the numbers that we're displaying here, but we've had a lot of teleconferences and we talk a lot about a lot of different topics. Most of the things that we talk about are specific products, but we do have a fair number of general topics that we discuss, and we'll see some examples coming up. This slide shows the number of pediatric cluster teleconferences that occur per year. 2007 was a half year, and this goes all the way to the end of last year, 2021. The red line in the middle of the slide shows you where our scheduled monthly teleconferences would be, and then everything on top of that is an ad hoc. So there is a possibility of going above the scheduled 10 conferences per year and having ad hoc discussions if timelines require that, or if there's a specific reason why we need to have frequent or urgent meetings. And you can see in the year of the pandemic that the pediatric cluster was very prolific. All right, so here's the promised slide about demystification. How do we add topics to our agenda at the FDA? At least I need to clarify that this is just for us, although any regulatory agency that participates in the cluster may request a topic. So at the FDA, usually topics can come up at the Pediatric Review Committee meetings when you're having detailed discussions about the pediatric development plan. Anyone from any of the participating agencies can request a topic, and sometimes sponsors will approach their respective FDA review division to discuss the product at the pediatric cluster. They can also approach EMA, although I'm not so certain about what the mechanism is on the EMA side. And it's ultimately at the discretion of the agencies to decide if a discussion at the pediatric cluster would be helpful or not. And that's another avenue by which sometimes we'll have topics added to the agenda. And then the way we view our topics are really like in one of two buckets. Either it's a specific product that we're discussing, or there's a general issue. The pediatric cluster does have an external face, and its external communications are managed by the Office of Pediatric Therapeutics, although it may undergo review and clearance in other centers and divisions in the FDA. The most, I think, the neatest, nimblest, and most useful tool is the Common Commentary. This process was started in 2012, and the Common Commentary is a very high-level summary of a discussion that occurred, and it will summarize FDA's and EMA's thinking, places where we agree, places where we may not be able to agree based on our differences in scientific thinking or our different legislative processes and requirements. And then if it's a specific product that was discussed and the Common Commentary was written about that, then that Common Commentary is shared typically by OPT with the sponsor in a confidential communication. And it's really considered to just be an informal, very nimble feedback on the pediatric development plan, and is in the interest of attaining harmonization for a global pediatric development plan. The next way that we communicate externally is with action items. These are also very high-level points that summarize, what FDA will share with sponsors that their product was discussed. Sometimes EMA shares the action items. This was started in 2018. And this was really at the just in the interest of transparency and at the request of industry to just communicate that your product was discussed at the pediatric cluster. Then the last way that we communicate externally is really to be a platform or a springboard that allows further development. So a conversation may begin at the cluster, and it may end up in a joint guidance written by EMA and FDA. For example, the Gaucher's disease guidance. And also a topic may come up, and it may become apparent that this requires more in-depth discussion, and that may end up in a workshop. And an example is the inflammatory bowel disease, gastroenterology, regulatory endpoints, and advancement of therapeutics workshop. And I think that is has had more than one iteration. So this next slide is probably the most interesting slide to this audience. It's the crux of sort of the neonatal part of it. Where does that come in? So the pediatric cluster is also the forum that international regulators use to discuss issues in neonatal drug development. And I have some examples for you. So in terms of information exchange, OPT shared INC neonatal drug development tools and work products at the pediatric cluster. And an example is the assessment of long-term neurodevelopmental outcome following trials of medicinal products in newborn infants. And we discussed a little bit about that this morning. It's also a forum where we can discuss issues that arise in industry-proposed neonatal clinical trials. And this could be a specific product, or it could be a general issue. And one example is a discussion that we had concerning the feasibility of conducting trials in neonatal seizures. And that discussion then led to a proposal to reconvene the International Neonatal Consortium Seizure Working Group. And you can probably guess who was involved in those conversations. In the past 5 years, we've had other examples of discussions that are regarding neonatal drug development. And that includes drugs to treat spinal muscular atrophy, which of course is very pertinent to a neonatal population, and products that are proposed to prevent chronic lung disease in premature neonates. And we also heard a lot about that this morning. So with that, this is a resource slide for those who may be interested in learning a little bit more about international pediatric drug development. And this last slide here just shows the structure of the international portion of the International and Ethics team in the Office of Pediatric Therapeutics and our contact information. So thank you very much for your attention.
Yes, dear colleagues, sorry, it's me again, but still I would like to thank the organizers for inviting me and to be at this brilliant meeting, and, you know, the first meeting since years. And these years have also had an impact on our neonatal guideline, unfortunately. So, as you know, as Inc. had provided some input on the concept paper to revise our really very old guideline, it had to be postponed during to, how do we call BCP, business continuity planning. So first it was Brexit and then COVID, and now we have to be very careful with our resources, apparently, and the good thing is that we are now really getting together with the PDCO and restarting this exercise, and then it will follow the usual way how our guidance or guideline development works, and we will have a public consultation, and obviously we will be counting on your support at the time. And otherwise, I mean, I don't have any specific neonatal regulatory update, but more, let's say, general update on in the pediatric space, which also includes neonatology, obviously. And this morning, yeah, every time I hear regulatory burden and regulatory constraints, it triggers several thoughts. And one thought is that we might have not yet done sufficiently initiatives to learn from each other. So one, obviously, and one, I mean, EMA sees this. I mean, we have a pediatric oncology space and the neonatal space where, you know, this multi-stakeholder approach is really being seen as flagship initiatives. And again, we are learning from our pediatric oncology space. And this space is actually just developing an educational program with placements with industry partners, but also at EMA for 6 months to not just sit around, but do a project and learn about regulatory and strategic science. I mean, if you see all these boxes, it really looks like a burden, but we need to come to a situation where it's seen as regulatory support. And obviously, a regulatory burden is also a good thing if it comes to high quality standards, and this is clear, as we have discussed. So after we have just seen the development, and as you might know, our pediatric legislation in the European Union is being revised at the moment. And after 10 years, there was a report and a big workshop, and benefits and room for improvements were identified. And on the more, let's say, administrative side, we really got the message, you have to become leaner. You have to make sure that we don't waste resources on all sides. And I just wanted to point out two main aspects here. One is the so-called evolutionary PIP. So a PIP is a plan, a pediatric investigation plan, and not a protocol. So here we have been also working with industry stakeholders to try to also see in our so-called key elements. Key elements are those elements which define how a study later on should be, which main elements a study should cover. And an example is we had several boxes. Regulatory people like boxes and tick boxes. And so we had several boxes for primary endpoints, then a huge list of secondary endpoints, exploratory endpoints, and so on. And here we are working towards a simplification. What is actually to define more study concept? What is the purpose, the key outcome of a study? Obviously, if in certain areas it is clear what we're talking about, yes, of course, we will also include primary endpoints and define them. It's just also to encourage, as you have seen on the slide, we ask for a PIP rather early, that you do come along, and even if you don't have the evidence to show what you are going to do exactly in neonates, we don't have to invent some placeholder studies. We can then agree, further evidence has to be collected, go to multi-stakeholder meetings, have a workshop, go to scientific advice, and then come back when we have sufficient evidence to set up and agree this plan. So this key element approach will apply to all PIPs, whereas this evolutionary approach, we will start a pilot first, and there we have a restricted set of cases which we foresee, because there is, after so many years, there is already in many areas enough experience to actually agree the plan at the time. For real world evidence and data, as I have mentioned yesterday already, also regulators are gearing up and trying to learn how to use regulatory world data and evidence to support their own decision making. At EMA, we have several ways or envisage several ways, and have started with first pilots also with the PDCO, albeit not in neonatology yet. So here we can either do it in-house by having access to certain databases, and we are just working on getting access also to hospital data, I believe at the moment French and UK data. Then in a framework contract, EMA can also contract studies. So they can have a certain research question and ask a study to be done. And finally, we have this famous Darwin network, which will be developed as a data network and service center, where also, off-shelf studies can be conducted, but also more complex, or as they define it, very complex studies. But that is all in development. And here I might see, having listened to your presentations this morning, I wonder whether it's also possible as a regulator to interrogate your data. I mean, we are fascinated by how Inc. has collected all these data. We will be interested in how they will be used, and honestly, if there are possibilities also as a regulator to, start with easy questions first, that would potentially be an opportunity. It is, we also try to provide regulatory support. So when applicants come along, it's really to tease out where, for a pediatric investigation plan, for example, where should we go to, which age groups, or in neonatology, what is actually the prevalence of a certain condition, not just from literature, but real at the moment. And that will be for sure crucial. And with this, I would like to thank you for this meeting and being here, and remaining good meeting. Thank you.
Hello, I'm Hide Nakamura, and I once was a reviewer at PMDEC. It's a former body of PMDA. So I visited FDA and EMA a while ago. And Junko Sato or Michio Sakiyama was supposed to be here, but they couldn't make it because of a conflict of schedule. I assume that's why it was not me to kind of compliment or whatever. And Michio contacted me and said they're trying to get funding for the trip to Inc. next year for at least for 2 people. But come on, if you can make it in Tokyo, that would be better. Okay, so let me do who I am. Like, I'm a pediatrician and pediatric pharmacologist, and now I'm chair of Committee on Pharmaceutical Affairs of Japan Pediatric Society. That's a counterpart of AAP, American Academy of Pediatrics. And we have, we get funding originally from funding agency, AMED in Japan. And now we're getting, and that's followed by funding from Ministry of Health, Labor and Welfare to set up JPEDNet, Japan Pediatric Society Drug Development Network. It's not a real network to conduct clinical trials. It's a category 3 network, just registered for EMPREMA. So we are like a group of specialists. So what happened often in Japan is even industry want to do clinical trials in Japan, or the domestic company want to do clinical trials in Japan. They don't have much experience and they don't know who to talk to. And if they go to 3 different people, they give you different opinion. Like, oh, what do we do? So what we're trying to do is we have working groups from different subspecialty, like pediatric cardiology or neurology. So we have about 70 different subspecialty areas agree to set up working group anytime. So anytime companies want to get advice or they want a recommendation for sites, we will set up a working group. I'm trying to pick out the best ones in the country. And also with research nurses, research coordinators to give best advice. And what we're doing right now. And in terms of PMDA, PMDA has pediatric working groups, but it's not like EMA or FDA. It's more like a virtual network. So it's what they call longitudinal activity inside PMDA. So people from different divisions have a working group. So maybe pediatrician, biostatistician, and pharmacist from different divisions. So it's I'm hoping that, well, I should say, so we wrote a reflection paper or recommendation with a former chair of the Committee of Pharmaceutical Affairs, and also Junko was one of them. And one of the recommendations is to make the pediatric division the real body inside PMDA, so that the pediatric team can give better advice or get good hold of the pediatric activity through PMDA. For now, I think, and correct me if I'm wrong, because for this pediatric cluster, PMD is trying to do the best. Like there's a lot of joint guidance, and also there's a guideline based on the discussion on pediatric cluster. And I think one of them is including teenagers in adult clinical trials in some disease areas. And another issue is we don't have regulations to mandate pediatric drug development in Japan, not like EU or US. So companies tend to start drug development later. And unless company consult with PMDA almost at the same time as EMA and FDA, they can't bring up that specific topic on pediatric cluster because there's a confidentiality issue. And I'm glad to hear that at EMD, sports cells can request to bring up the issue to pediatric cluster. And I think we hopefully PMD accept the same request. Problem is that they don't have that much because there's no consult about that particular trial. They can't actually bring it up for discussion. And the reason why I'm here, like I think I visited, I'm here at Inc. from the day one. I was the only one. And then for next time, I talked to Satoshi. You know, if this is so important for Japanese neonatology, so you should come. And he agreed to come. And he said, okay, this is important. So she started bringing more people. So that's why, you know, there are like awardees from Japan too. And I don't think there's any neonatologists in PMD, or maybe a couple of them. It's like, even though we go for a consultation, we don't see much appearance of pediatricians. Well, some of them do, but not many. So I'm also hoping to not educate, but hoping to bring in more pediatricians to PMDA itself. And our network is hopefully the network of Japan Pediatric Society for now is kind of complementing the expertise. They don't have too much, and also for any industries. And one good experience is that there's a global trial ongoing right now in Japan, US, EU, and Japan. And it started here at Inc. One company started to contact me and Satoshi about doing global trial together. So we're at the phase 2 meeting together. And then they started trial together. So I'm hoping that and next year, if we can do that in Tokyo, I'm hoping to bring more PMD people and neonatologists, and hopefully you guys from companies can start trial more. And I understand that even though global team try to convince Japanese team to come to Earth or PMD, sometimes they're hesitant. So I want to rectify the problem. So having Inc. meeting in Japan would make a big impact. One of the reasons why Junko and Satya's supervisor, somebody in between Executive Director Yasu Fujiwara, he's a good friend of mine, and Junko, there's somebody who's probably, in general, they understand it's pediatrics important, but neonatology, I think, I know him very well, and I think what he had in mind is, oh, wait a second, you know, we can't even do pediatrics. Why neonatology? But I think it's vice versa. Unless we do neonatology, we don't understand all the, you know, it's the beginning of human. So it's very important. So I think that's something we have to really have Japanese people understand through, if we can make it in Japan. And well, I think, thank you for this opportunity. And well, I'm not really PMD, from PMD, but I hope you guys understand how important it is to drug development in Japan also, because we have children, neonates in Japan too. Thank you.
So thanks to my colleagues. I just have a couple of quick questions, things that we could discuss, either for Sarah or Hide. Would someone like to talk about the workshop that FDA has been conducting with PMDA for the past couple of years? And you can, I think there are microphones there. So FDA and PMDA have had an annual workshop since.
That's a PMDA course. Like they have an Asian training center for they are regulators. PMDA is trying to, yeah, that's something I missed. I wrote it in here. Oops, I missed. Okay. There's a Asian training center at PMDA, and they're trying to educate regulators in Asia And also, there are some some regulators from other developing countries as well. And FDA folks are always there to support PMD. And I think I was there for a couple of times to talk about pediatric pharmacology issue too. And it's a very good interaction. And FDA is helping PMD a lot. And I'm hoping that PMD in the future can do it all by themselves. For now, it's still, they have limited number of pediatricians. And I think that's a very good collaboration.
Yeah, it seems like it's been good for information sharing.
Yes, I want to also say that I was also thinking about the PMDA and FDA workshop on pediatric regulation. I haven't been part of too many of them. I had listened to the last one, which was virtual. And I thought it was a very, very good platform for many developing countries were represented that I noticed. And there's there's a presentation of information, and there's breakout groups to go through cases and really work through some of the pediatric regulatory issues that, we may have a little bit more experience working through, but honestly, with every application, we work through those issues as well. And that's a yearly event. And that's also a big part of what my particular team in my office does. Donna Snyder, and it used to be Kathy Lee. Now it's Suzanne Molly taking care of that. Thank you.
Thank you. If there's any questions from the audience, we certainly have time. I just wanted to ask a question to Ralph. Do you, I think folks here are aware of the new ICH pediatric extrapolation guideline. Does EMA foresee any changes to the approach to neonatal drug development as a result of the extrapolation document?
Unfortunately, having a document doesn't make it happen necessarily, as you know. And doesn't make it easier. Apart from the situation in neonatology, we are even, this is also a field where we are learning. So this is, I mentioned it also as an example where, where we have formalized something which we sometimes did implicitly and really to think about target population and reference population. However, unfortunately, as we have heard, with different diseases, at the moment, this approach is not used from our side in the neonatal population. Perhaps as we learn more, and we might see or might learn that there are more overlapping situations, yes, but at the moment, we are not so optimistic, and the same is a little bit for modeling and simulation. So this is really, really unfortunate for this group. Yeah, perhaps the next tool might be it with real world data. Yeah, that is what we're looking forward to.
Any other comments on that? Klaus, do you have a question?
Go ahead. I'm not from PMDA, so from academician standpoint, that 11A is, well, it's still kind of, there's a lot to experience to make it better. But for many Japanese, they don't understand the clear concept of extrapolation. So even to the people of Clinical Research Center or Boa Center, I translated this case series slide to Japanese and try to have them understand what the idea is. And I think for many Japanese people, it's nice to understand what extrapolation is, how they can utilize it.
Right.
And even among the reviewers, they may have different idea of what extrapolation is.
Right.
So I think it's a very good.
We do have some opportunities in neonatology around, particularly anti-infectives, not entirely extrapolatable, but, some of the conditions and some of the indications, we can use some extrapolation of efficacy.
Exactly.
But, so far we haven't been able to figure out how to get around understanding dosing in neonates, proper dosing for exposure and safety. So
Yeah, so first of all, thank you. I don't think that there's any other forum where this conversation could happen. So this was fantastic. Ralph, I want to get back to one of the comments that you made about maximizing the utility of the data. One of the things that we have heard loud and clear at the meeting, and it clearly came out as an action item that we will take back to Tucson and deliver on that, is the need for Inc. to have an articulated research agenda for a time cycle. We kind of define 3 years as a sweet spot. We'll have that conversation. But one of the things that we also heard from our industry colleagues is precisely that question. How can we not just focus on the demonstration projects that we have defined, but identify other demonstration projects with the data that we can leverage? And I think not just from the standpoint of what you see as a regulator as a need, but what the industry colleagues see as a need. And one thing that I would love to see is the matching of those things and start with the common denominators, because I think that could be a pretty important prioritization exercise. So if you guys are committed, we're committed, and we can make that happen and capture that in the research agenda.
Great.
You're nodding, so.
It's not only up to me, obviously. From my personal perspective, yes, but I mean, it's good. Like in every organization, we need this discussion, but as I said, I'm optimistic for that, and we need to push.
Thank you, Mary.
So I'm curious about the impact of tools that come out of INC. And, I'll use an example, the Neonatal Adverse Event Severity Score. How is that being integrated in a regulatory pathway forward? What discussions do you have? Has it been shared or recommended to sponsors?
I'm happy to share, but I would let others start. I will say at FDA, we literally hand it out to sponsors. I mean, we use it. We recommend it to sponsors. We capture some information about our consults, our neonatology consults. And we just recently thought, thanks to one of our colleagues who's here, that we should start capturing how many times we actually hand it to sponsors and say, we need you to capture good systematic capture of these adverse events. And here's a tool that you can use. So we certainly use that. I will also say that the seizure master protocol has been handed to investigators from the Division of Neurology. And those are just two of the tools that we promote internally all the time. I don't
Jerry?
Yeah.
Sorry. I just from the perspective, from the CRO perspective, we work with sponsors all the time in the neonatology space, and we're incorporating them into the protocols, the adverse event scoring.
Yeah.
Critically important. So I will have numbers maybe next year on that, but definitely including that in all of our kind of responses.
And I'm fairly certain that we recently recommended the blood pressure measurement document as well.
I just wanted to follow up on Klaus's comments because Ralph had brought it up. I think we want this database that you've helped fund to be an extremely valuable resource, not just to us, but to you. And, just scratching the surface, looking at background rates, this is the only, at least as far as I know from the neonatal perspective, global database that exists. Certainly individual sponsors may have sites, but this is, almost 500,000 infants from a variety of different countries. And so just looking at background rates of adverse events. So when you get all these events coming in from trials, is this significant? Is this not significant? As an IRB chair, that drove me crazy because I never understood the context. When Sunny presented her data today, what struck me is when she said the control group was different than what they had anticipated. And I'm wondering if we couldn't be helping some of these large multi-center trials before we jump in to say, well, this is what we anticipate the background rates to be, but could we look at your data set and see, using real world data, is that really powered appropriately for what we want? And can we not only power it for the short-term outcomes, but can we power it for some of the follow-up as well? So I think, and again, that's just off the top of my head, I think there are unique opportunities to work with the different regulatory agencies who can then query that data set with questions that come up with respect. And I'm hoping maybe that allows us to do more extrapolation potentially. And, you know, I was encouraged when I heard Lynn or Yao and others saying that maybe we can even extrapolate in certain conditions for safety as well. That's a very novel concept. And then can we go back to the data set and can we validate some of these thoughts? I think that would be extremely useful. So we'd be anxious to hear from the different regulatory agencies to say, you know, we have a question or we'd like to work with you on this. And that, I think, was what you were proposing, Ralph. And I think that would be very helpful back and forth.
Great. Any final comments? It's been a pleasure to be here. I think we have a lunch break. And since we're starting the lunch break early, I don't know, Kamalji, you're the director. You're in charge. Thank you.
We're happy to. Unless anybody has any other questions, we're happy to declare lunch 5 minutes early. But we'll convene at the same time, 1:45 p.m. Eastern, to go over the last sessions of the day. That is the work group updates of all of the Inc-related projects that have been going on. So thank you. Hi, everybody. Good afternoon. So we're going to get started with the last session of this meeting. That is the work group updates. These are going to be very short 15-minute updates for all of the INC work groups and everything that they've accomplished thus far. And the very first one that we're going to get started with is the hemodynamic adaptation work group. And for that, I would like to welcome Dr. Heike Reb, who's the professor of perinatal medicine at Brighton and Sussex Medical School in UK.
Okay, thank you. Yes, good afternoon, everybody. First of all, I would like to thank C-Path and Inc. teams for organizing this wonderful meeting. It was so nice to be back in person. And I'm here on behalf of the hemodynamic adaptation work group. So I thank all the members who have supported the group during the past, I think it's 5 years now. It took a bit longer than anticipated because of COVID and everything. But we are getting there. So for those who do not know, when we started the group, we first defined the questions, the pertinent questions which should be answered. And there were 3 of them. So there were Delphi rounds and very intense discussion. So the questions we were tackling was, what are the observed ranges of blood pressure by gestational age, weight, and postnatal age in babies who have not received any modifying treatments? So to get near to normal values. What other factors influence the blood pressure and how? So especially around maternal factors, medication, and perinatal factors. And the third was, what are the recommended measurement methods? So that was done by means of doing really extensive systematic reviews on all these 3 questions. And we then tackled them in the reverse order. So we started with question 3, which has been published as a systematic review in Journal of Pediatrics. That was at the end of 2020. And that has been really a very successful publication. As you can see on this slide, the metrics are all above the 90th percentile for comparable publications, but also most importantly, apart from being used, it's at the regulators quite a lot in studies to be done. It has also been picked as one of the 3 CPD activities for pediatric nephrologists by the American Academy of Pediatrics. So it's very much read and also shared in their journal clubs, so that it really gets a lot of attention through this matter and has been cited already quite a lot as well. The second publication focused on antenatal and perinatal factors influencing neonatal blood pressure. Again, we also identified where are actually the gaps in knowledge and where where future research needs to be done. And that has been published last year. It's already started to be cited and read quite a lot. We get the alerts through the ResearchGate. And now we are currently focused on the last paper, which we have planned, which is about observing ranges of blood pressure by gestational age at birth, birth weight, and postnatal age in infants. And this is one of a small graph, but just to give you an overview of what kind of figures we will be using. So this is a graph on mean arterial blood pressure by postnatal age in preterm infants. And that's organized by gestational age. And as you go through the gestational age, also by age after birth. So you can see clearly that the blood pressure is indeed going up. And but there's a wide range. So yesterday we had a very good constructive meeting of our working group, and we were able to talk about what would be the key messages, which you also need to submit with a manuscript. So a manuscript is nearly finished basically, so it's well written. Just working on all these results, and I'm very grateful to Stephen Bremner, as a statistician at Professor of Statistics at my medical school, and also Ash Santu at Vancouver University, who have done really a lot of work in getting this into a format which is then useful for the readers. And so the key messages which we have now drafted are that neonatal blood pressure values increase with gestational age at birth, not so much with birth weight, but also with the postnatal age. But the acceptable blood pressure values in neonates who are not on modifying blood pressure treatment can fall within a very broad range, probably a broader range one would normally have thought about, or what we normally use. And quite often the observed mean arterial blood pressures are below the numerical equivalent of the gestational age at birth, which we are currently using, but which never proved or proper study really since 40 years. So I think we have refuted this myth now. So that's one of the main results of this paper. As you know, with the other working groups, there's a paucity of data in the first weeks of life. So we've suggested analysis of multi-center patient data is needed to provide reliable, collatable, and thus informative blood pressure ranges. And I'm very happy to hear from Kanwaljit that is already in the real-world data collection. So I'm very happy about that. And we will make also some recommendations about future collaborative research, which then can use these blood pressure measurements at endpoint for future drug development studies. So thank you very much for your attention.
Thank you, Heike. The next one up is the neonatal pain work group by Dr. Rebecca Slater from University of Oxford.
So many thanks to the organisers for organising this meeting, which has been really fun. And I'm delighted to share some of the updates from the pain working group. This is a relatively new group, and so I can just share the work that we've been bringing together over the last few years now. So, but before I start, pain is fundamentally protective. It prevents us from damaging limbs. It teaches us to look after ourselves. But it's simply not what we think about when we imagine children going home after they've been born. We imagine them in the care of their mothers, fathers, siblings, and others who provide these functions. But it's just simply not the case for the 1 in 7 children that are admitted to hospital after birth. And for these children, they'll experience an average of around 10 painful procedures a day. And in the youngest and the sickest, this can go up to 50. And for these procedures, pain relief is simply rarely provided, even though we know that there are long-term consequences. So the Pain Working Group had a mission, which was to create a roadmap to be able to find better ways to accurately and reproducibly measure acute pain in neonates that could be capable of being used for neonatal analgesic drug development. And the working group is here, and it's been wonderful to meet many of you in person over the last few days. And indeed, if anyone else is interested, it's a big open club, so please come and join us. But we've got representatives from C-Path, from industry, from the FDA, and from academia. So just to kind of give you a brief timeline of this project, it was in May 2019 at the 5th annual INC scientific workshop, where we discussed the importance of having better endpoints for evaluating pain in children, in neonates. And this was followed up with a really helpful CEDA meeting where we were able to look and see what the evidence gaps are in terms of the most appropriate way to measure pain for this population. This was really timely because it came out at around the same time as the Lancet Child and Adolescent Health Commission, which was about delivering transformative action in pediatric pain and really did set the call to action and priority setting for this field. And further followed up by a 2-day workshop where we were able to gather more evidence about the current state of the field. And so it was in March 2022 that the first working group really commenced and started pushing forward this work. So what are we trying to do? Well, we know that there are numerous painful acute procedures that are happening while babies are in hospital. And there are a whole range of different analgesics which can be used to try and treat this pain. And of course, because babies can't talk, we're reliant on a whole range of different outcomes in order to try and understand what's happening in the infant's nervous system when they're experiencing pain. So we can measure reflexes, change in heart rate, behaviors, their physiological instability. But importantly, we can also measure activity in the brain. And this is important because this is the organ where pain is happening. So if we want to better understand the perception and not necessarily be dependent on looking at downstream responses and motor activity, we can start to consider whether brain activity can be used to create objective measures of acute pain in babies. And so we have a measure. And through a number of different consultations and thinking about which best approach to go through, we've been considering the use of EEG and looking to see whether a pattern of noxious evoked brain activity that can be recorded in response to acute nociceptive procedures can be used as an objective outcome measure to measure the efficacy of analgesics in this population. And so to this end, we've decided to do a look at an individual patient data meta-analysis to ask the question whether this noxious pattern of brain activity is effective, and then to look at a number of different features associated with this measure, such as the validity, reliability, and other important constructs that would be essential in order to have appropriate measurement properties for us to take this forward. When we've got some confidence that this works, it's exciting that one of the groups in UCL recently released a large amount of their data, and we were able to compare the patterns of brain activity in children from 29 up to 39 weeks across these 2 independent sites. And you don't need to know anything about what these patterns mean other than to see it looks like we've almost drawn them on top of each other, which suggests that there's a really high possibility that these are reproducible across these independent data sites and that we may be able to use them. Furthermore, these measures have already been used in a whole range of clinical trials, and I've given a few examples here to measure the efficacy of a number of different pharmacological and non-pharmacological interventions, including sucrose, paracetamol, morphine, tactile stimulation where the mother or father can stroke the baby prior to these procedures, and local anesthetics. So the start of this project was to do a scoping review to identify all of the studies where noxious evoked brain activity has been used in response to acute nociceptive procedures. And the scoping review, we literally took 2,600 or so different studies from databases and clinical registers. And then through the normal PRISMA processes, went through these reviews and finally identified 43 studies in databases and 14 studies in registries where we have these EEG responses to this acute nociceptive stimuli in this population. Brilliantly, the data is across 3 continents and 9 different countries where it's being collected. Excuse me. And we can see that there are a number of different data families that we've identified. And by that, we mean the same authors that have contributed to a range of different studies. Much of this is my work, some of the work in UCL. There's a lot of work in the US and Canada as well as other countries that you can see here. And so of the data that we finally summarized, there are 57 different papers where we're able to see these procedures, which include heel lances, venipunctures, and vaccinations in the population that we're interested in, where in these studies, a whole range of different interventions have been looked at. And in addition, we can compare some of these measures to some of the more classically recorded non-EEG measures, such as the clinical pain scales that would currently be in use. And so through doing this and using the COSMIN guidelines in order to ensure that we've got the appropriate measurement properties to take this measure forward, we can now look to see whether it can be an appropriate pharmacodynamic response biomarker. And we can start to look at the inter-rater and intra-rater reliability of this measure. We can take all of this individual data, bring it into one analysis, and look at a number of other features related to its interpretability and indeed the validity. And so having now done this part of the scoping review, which is the first stage of the individual patient data analysis, we're at the stage where we are able to register this work with Prospero and submit it to BMJ Open or systematic reviews in advance of really taking the raw data and asking these primary questions that I've just presented to you. And it was very exciting in the work group meeting yesterday that the final comments related to this have come in, and we're ready to take this to the next steps. Listening to you all talking about how you manage these big data sets slightly terrifies me. But nevertheless, I'm optimistic that the pediatric pain world are just going to give it to me when I smile at them or something, but we'll see. But we've probably got another year or so of collecting and curating this data so that it can be then analysed to ask these important questions. So finally, I'm just going to end this talk with some optimism about where we're going. The Lancet Commission on Pediatric Pain really did set a set of challenges for our field to make pain matter, to make it understood and visible in this population. And the work of the INC and the bringing together of the different groups here featured highly in this commission. And as a direct result of that work, the International Institute for Health and Care in the UK, so our NIHR, have put together a specific ring-fenced call to look at pain management for children and young people, where they're specifically talking about the importance of acutely painful procedures in neonates as one of their priority areas. And this is a call that will be coming out now, so we'll be putting in an application with many people in this group to really try and ask whether we can reduce pain associated with vaccination. At the moment, we often give paracetamol post-vaccination, and there could be strong evidence to suggest if we gave the paracetamol before the vaccination, that this could reduce the pain-related activity associated with this procedure. And just, oh, I think I've skipped a slide there. Maybe not. And so this group of people have come together to put this call together, including many people here. We're very excited through initiatives like this, where John Vandenanker, for example, is now going to come to Oxford for a year under the Newton-Abraham Visiting Professorship Scheme, so that we can really link some of our PK and PD knowledge and bring it together into one centre. And it's through these kind of initiatives that these people have been able to come together in this way. And so hopefully this pilot data that you can see here, which is using the measures that I've been describing, that suggests that when you give paracetamol prior to an immunization, the magnitude of these responses reduce, we'll be able to see this in a large-scale clinical trial, ideally funded through this NIHR call. And so finally, just because we've been speaking so much about the importance of parents in the neonatal unit, I'm just going to tell you about one more piece of work where these measures are currently being used. So in our PETAL trial, which is a study where we're looking at whether parents can stroke their children for about 10 seconds prior to a painful procedure, can reduce the noxious evoked brain activity. And we've currently recruited 98 babies. I hear one was just recruited this morning. And so we're really in the final stage of this, using these kind of outcome measures. And simply put, if it's true that activating some C-tactile fibers on the skin for 10 seconds prior to a nociceptive procedure can reduce the pain and cause this inhibition, then there's simply no reason why we shouldn't be doing this for every single child globally. And so I'm very much looking forward to be able to share the results of these outcome measures. But in order to really be able to use them, we need to get them endorsed and run through all the complexities of really establishing that they're valid and appropriate. And certainly as a group, I think we've got a long way to being able to achieve this. So thank you very much. And I very much look forward to telling you the next updates from this working group.
Thank you. And next, I would like to welcome, excuse me, Dr. Lewis from Sick Kids Canada.
Hello, everyone. As mentioned, I'm Tamara Lewis. I'm from the Hospital for Sick Children. I'm representing the safety work group for INC, and this is just a list of current members. And the goals of my talk today are to give you guys an update about the neonatal adverse event severity scale, hopefully engage stakeholders and potential next steps for the severity scale, and then really not necessarily to gather input, but to encourage everyone in the room to think about input on the next projects for the safety work group. So we've heard a little bit today about the neonatal adverse event scoring system, and we very early on in INC recognized that a major barrier to regulatory drug development in neonates is that these children are often already very sick in the ICU, and it's difficult to use traditional adverse event severity scales to understand the severity of adverse events, or even to recognize, is this an adverse event in this population or not? So Thomas and the group at Leuven really led the initial development of the NAESS, and it was published in 2019. And here in the figure is just an example of the generic scale on the top panel in A, and then an example of an AE specific scale on the bottom in panel B. This is a scale specifically around infantile apnea. And the scale actually includes multiple different AE specific tables like this that can guide the PI or the research coordinator or the monitor about an individual adverse event and how to scale the severity. But we also recognized that in order for the new tool to be accepted into broad use, we would need to perform validation studies. So the safety work group decided to take two different approaches. First, we performed a retrospective validation study using existing safety data from a neonatal randomized controlled trial. And that was actually discussed by Dr. Wade at last year's INC meeting safety work group and published in the Journal of Perinatology. And then our second step was to perform a prospective validation study using brand new safety data collected during both research and clinical care in 4 different NICUs led by Thomas. So very, very briefly, the results of the retrospective study. So this study used real-world clinical trial safety data from a neonatal RCT led by Dr. John Davis. We took 60 adverse events with a range in severity and randomly assigned multiple adverse events to 12 different reviewers. The demographics of the reviewers are displayed here in the table from the paper. So at the end, we had 240 adverse event scores that were available for analysis. In the retrospective study, we found that in the hands of novice users of the NAESS who only had access to historical AE data on the original case report forms, the tool showed moderate reliability. So the intra-class correlation, or the ICC, was 0.63. And what we found is that serious adverse events had a higher ICC than non-serious adverse events. And we speculate that is because of the much greater amount of detail provided on the case report forms for SAEs versus non-SAEs. What we also learned in the retrospective study is that the two types of adverse event tables in the tool, I'm going to refer to them as generic versus AE specific, were not used as predicted by the 12 reviewers. So the researchers kind of had a sense of how the reviewers would use the tool, but there were some unexpected results. So for example, there were cases where the reviewers inconsistently used generic versus disease-specific NAESS tables. So for pneumonia, tracheitis, and UTI, we don't have a specific AE table, but instead of using the generic table, which is what we expected reviewers to do, reviewers often used other AE-specific tables such as culture-positive sepsis or culture-negative sepsis. In addition, patent ductus arteriosus has no AE-specific table. The study team predicted that the reviewers would use the generic table, but instead the reviewers often were drawn to other AE-specific tables, such as respiratory insufficiency or edema, when the actual AE was a PDA. So the data from this retrospective study performed really helped us design the prospective validation study and helped guide which questions we would ask. So for the prospective validation study, we designed a two-phase protocol and deliberately included a structured teaching tool between phase 1 and phase 2 so that the novice users would get structured training around how to use the NAESS. So in phase 1, 30 adverse events were recorded, and these adverse events were identified in the NICU in a prospective manner, and they were graded within a certain period of time, 72 hours from the AE event, based on personal evaluation by 2 observers at each site. There was a training phase where both observers followed a structured training module on the use of the INC NAESS. And then in phase 2, an additional 30 AEs were prospectively recorded and severity graded based on the INC tool used by the 2 observers. So in phase 1, we used the CTCAE, which is I would say, kind of non-neonatal specific adverse event severity tool. And this kind of gave us what I would call a baseline interclass correlation, which we hope to use as a comparator arm to phase 2 when we would be using the NAESS in a trained cohort. The 4 sites were international, and the sites and the PIs at each site are listed here. Each site individually underwent IRB approval and was given access to the central REDCap system where all of the data from the 4 sites was collated. And each site had 2 naive reviewers who had no familiarity with the NAESS tool. Interestingly, in this prospective study, it was mostly clinician and like physician investigators who were the reviewers and less research coordinators and regulatory people. So this is an example of the range of AE-specific tables that are in the NAESS tool shown in the figure. And with 60 adverse events collected at each site, we had a goal that no AE was repeated more than once in each phase. We had a mix of generic versus specific AEs. And then our Critical Path Institute biostatistician, Kurt, helped perform the data analysis. Inter-observer variation was measured using interclass correlation. So what we hypothesize is that if the NAESS tool is superior to historic AE tools for neonates, we would expect the ICC to be higher in phase 2 versus phase 1. So the results I'm going to share with you today are not final. Thomas, Kurt, and I are still very much in the data analysis phase, and so things may change a little bit, but I wanted to share some preliminary results. So the phase 1 ICC is 0.66 and the phase 2 ICC is 0.69. And in order to compare the 2 phases, the confidence interval is shown. And the interpretation based on our preliminary analysis is that the tool in phase 2 performs slightly better than the tool in phase 1. What's very interesting is that if you look across the 4 different sites, the tool had different performance. So you can see that in Kansas City and at the Canadian site, phase 2 clearly performed better than phase 1, meaning the NAESS tool had greater intra-class correlation than the CTCAE. But at the Japanese site and the European site, that was not the case. And what we found is that when we dove deeper into the data, it was really the generic table that was driving the worst performance at the 2 sites that did not meet our hypothesis. And so I'm not going to show you the site-specific generic versus AE, but overall for the entire study, you can see that when the reviewers used the AE-specific table, the phase 2 was clearly better than phase 1 for the intra-class correlation. But when the reviewers used the generic table, the phase 2 performed more poorly. And so this is really what drove the performance at the 2 sites that phase 2 did not improve. And so what Kurt, Thomas, and I are currently doing is looking at each site in more detail, looking at the generic versus the AE specific table. But really, I think this, if anything, is helping us understand that the true power of this tool is in the AE specific tables and not necessarily the generic one. So we as a safety work group are very open to feedback from the INC community. For potential projects, one thing that I think is a very good idea is to expand the number of table-specific AEs in the tool. And Kelly Wade and I were talking earlier that it may be a good idea for INC to set maybe like a benchmark of adding 8 or 10 specific tables per year. I also think we could harness the power of Critical Path Institute to create a formal teaching tool so that reviewers who are going to use the NAESS have a very well put together teaching module. Thomas created one in PowerPoint, but I'm sure that there's a much more luxurious way to do that. And then also, I think it's really important to think about how do we collaborate on adoption and more universal use of this tool by pharma and regulatory bodies. INC has really led the development of this neonate specific AE tool, but I think other ideas that have been percolating is that we could consider doing an analysis on multiple different data sources and write a white paper on the safe and reasonable number of study-related blood draws in neonates and infants, for example, in order to describe pharmacokinetics of a drug. And then we're definitely open to any other ideas. So thank you.
Next, I would like to welcome Jerry Baer to talk about the bronchopulmonary dysplasia work group updates.
No slides, and this will be very brief. I might back up here again. So the BPD working group was dormant for a couple of years and has been reactivated and has now begun to meet again in sort of in parallel with the ongoing real world data, real world evidence grant project. And so there are on the one side, these ongoing discussions about efficacy endpoints and how to determine the best efficacy endpoints in this space. And then at the current face-to-face meeting, we were able to have a discussion about what kind of questions might we be able to begin to ask of the real world evidence data sources. And so a couple of the things that sort of arose out of our discussions yesterday were one, to consider sort of a landscape analysis of what data we have, looking at, how many patients do we have that have long-term outcome data, either, somewhere in the 1 to 2 year range, that can be linked back to the NICU admission. So finding out, how many of those do we have? Where are they from? And then we can determine what variables we might want to look at, or whether we want the data to sort of tell us. And then we've also talked about asking some questions about some of the more severe BPD phenotypes and what are the antecedent factors. So this is really, just kind of getting revitalized. And so, please come and join us so that we can begin to use this fabulous data source to help advance BPD therapeutics. That's all.
And finally, I would like to invite Mary and Kelly for communications work group updates.
Hi, everybody. Hey, my partner in crime, who has been such a wonderful co-chair, has had so much fun at this meeting and so many conversations that she's lost her voice. So I'll be presenting today. Okay. So we're going to share with you an update on our messaging across stakeholders for Inc. We're going to remind you about the communication toolkits. We're going to talk about next steps for the MASH project, and then what the communication work group has decided to work on next. So during 2020 to 2022, our work group's focus was really on messaging across stakeholder groups for Inc. And, that was the key message development from the perspective of each stakeholder within INC. And we want to be able to communicate Inc.'s progress to Inc. and to the larger neonatal community. It's important that we really amplify and support the perspectives of all the stakeholders. And our goal is to promote a positive research culture for neonates so we can address their unmet medical needs. So this is a slide of the deliverables from INC, and congratulations, everybody. It's a great body of work. And where you see the red stars, those are where we have completed the key message development. And the text box there shows you all the different deliverables that have the key messages developed. And we've completed 3 this year, and we have 2 ongoing this year. So we have kept busy. And well, that's great that we have key message development, but the most important part is then that we work with the C-Path communication staff to put these into a toolkit. And there is a toolkit for each of our stakeholders group, again, so that we're communicating the progress, amplifying the message, and promoting the positive research culture. And this is to remind you, where do you go to find those resources? So you just go out to the INC website and you go to the resource page there, and then you'll see INC Communication Toolkit. And then you'll see it's set up for each of the stakeholders. So please be sure to go out there. And then the key messages are in many different formats for your use. One of them is the social media toolkit. So we have great graphics and images that can be used. And we have key messages for you to use, and they're formatted for whichever social media venue that you're going to use. It's really important as you have papers coming up, or you're reaching out because you're going to attend meetings and you're going to talk about the subject, to go out here and use the toolkit. Then we also have PowerPoint slides for each of the stakeholders' perspective on an INC deliverable. So if you're going to a meeting and you are going to talk about a topic that we're working on in Inc., all you have to do is go grab the slide and throw it into your presentation. We also have, and what you see on this page are the information one sheets, and you see one there on the severity scale, which gives a little more detailed information and but concise format for you to use as appropriate. The toolkits also have a draft email blast. So if you want to reach a large group, we've got it all message for you. Just plug in your key message and use your email distribution list. Then we also develop frequently asked questions for all of the Inc. deliverables, and they're set up in the same way. So how did this project come about? Why is this project important? And what is INC doing to facilitate the use of this deliverable? And how we view these frequently asked questions is that they're talking points for you, so that we all stay on a good sound message for the work that's at Inc. So please, again, on here, you just go out to the Inc. website, and across the tab, you'll see FAQs, and that's where those that tool for you is. And so now we're announcing today that the key message project, which we have been doing retrospectively for the manuscripts that are already completed. Now they're moving forward to be prospective. So in each new INC work group, within the work group, the key messages will be developed in the initial process. And really that only makes sense as you're working on a project, you know the target audience and you know what you want to communicate to them. Then the communications work group will still provide support and will be a consultant. And we're going to hope to make sure that all stakeholders are represented in a work group. But if we would come in a situation where that's not the case, then our work group will provide those stakeholders to you. Just a high level, I'll go over to this, what the new process will be. So when a new work group is initiated, the communication chairs are going to meet with their chairs, and we're going to talk about the process, and we're going to review the inclusion of all stakeholders. And then the work group chairs is the work on the left here. They're going to identify who within their group is the stakeholder lead to develop the key message for that stakeholder group. Phase 1 is brainstorming, so they'll just brainstorm the messages. Phase 2 is a refinement stage, and we have tools to help you go through that refinement. When that work is done, it'll go back to those work group chairs, and they'll make sure that the messages are consistent with what's actually in the deliverable. And then it'll come over to the communications work group, where we'll do the phase 3 testing, is when you reach out to more of the stakeholders and make sure that you get refinement. And we have about 35 members of our work group, and they're all very engaged. And then it goes back to the work group chairs to make sure that they approve any final revisions we've done. It goes to the Inc. leadership for approval. And then it goes to the C-Path communication staff to be incorporated into the toolkit. And here now, the goal is that we're going to have key messages available at the time that you go to a publication or presentation of your deliverable. So these are the work groups that we're aware of that we'll be working with this next year. We've already met with the lab values and the hemodynamic adaptation group. And the rest of you that you see your names on this slide will be reaching out to you. And then talk a little bit about what our next effort is going to be. And in doing that, I'm just going to kind of remind you where we've been and now where we're going. So initially we were a newsletter group, okay? And we're not a newsletter group anymore. In 2018 to 2020, we had that culture of research communication survey. And in the last several years, we've been sharing those results. We did a webinar. We have a publication in the Journal of Perinatology, which included a parent editorial. And happy to announce today that we actually got the news today that the nurse perspective, the additional survey results that weren't published in the primary manuscript, were accepted for publication in Advances in Neonatal Nursing. So as the group talked about what they wanted to do next, we really aligned on the need for education. And in the Journal of Perinatology article, the conclusion is that nurses and parents have suboptimal engagement and that there is a need for education to enhance their engagement. And so our group really wants to focus on the components of education that shares the importance of research in the NICU, and that how that research has been foundational to the advances that we already have for babies, but how we need to move forward to meet their unmet medical needs. And that there needs to be, one of the survey results was that nurses felt they weren't asked, but they also felt that they didn't necessarily have the optimal education to support their participation. So that's an area of a focus for us. And if you remember, in 2019, our industry colleagues provided a half-day educational workshop on drug development, and we would like to repurpose that information and use it as foundational nursing education. So we've reached out to those presenters and have permission to reuse those materials. We have support from INC to re-record that information because unfortunately the audio quality of those recordings is not very good, and it's not the type of product that Inc. will want to put out there. So we will be doing that work this year, and again, it'll be publicly available from the website. We also thought that the communication flow in the NICU was an area, and in that we're talking again about what's available and are we really promoting and embracing the positive culture of research in the NICU. And when Kelly and I talk about it, we just feel like maybe we haven't yet told the story well. So our focus is how to tell that story. So in our workshop yesterday, we decided that we're going to be rather ambitious here, and we are going to take on a social media campaign. So the details of that will be fleshed out and you'll learn more in the upcoming year, but we are very committed to sharing the message, telling the story well, and making sure that we amplify the impact of ink. We have a great communication work group, and you'll see here all stakeholders are included, and we are an international group. One of our newest member is Dr. Tomakati from Japan at Kyoto University. So we welcome his participation, but we also welcome other members. And, you know, we're taking on a challenge on a social media campaign, not necessarily pulling from our own strengths. So if like our industry colleagues have folks in that area who they might want to get involved in ink, we sure could use that membership. We have some great parents who are active in that space, and we met a new parent active in that space at this meeting. So we hope to capture you all, but we definitely welcome new members, and we want to thank our members. They've done a great work for us. So thank you all very much.
Thanks, Mary. Thanks, Kelly. We do have some time to allow people to ask questions from the respected members of the work group. So if you have any, happy to take some time to go into a Q&A session if you guys want.
I'll be happy to ask a question.
Yes.
A few comments first. First, I wanted to thank Mary and Kelly. You know, this communications work group has really found its footing, in my opinion, and watching it evolve over time. And it's really now operating with leadership, a single purpose. And I'm really delighted. I think we were all jumping for joy at this table here when we heard the paper was accepted. So my compliments and thank you to that group and to all the people who are really participating. I think the message needs to get out. As Wakako keeps telling us, the nurses are stressed in the NICU. They're taking care of more and more patients. There's nursing shortages. And when you ask them to do a variety of extra procedures, whether it's blood draws or other specimens, it's hard. And so educating them of how important that really is really critical. So my compliments to that. I'll say a comment also with our presentation in BPD. We're really excited, as Jerry said, about the prospect of using this large data set. Traditionally, we all looked at risk factors for bronchopulmonary dysplasia and then followed the kids out on a forward basis and found what the issues were at a year or 2 years. And we found that BPD wasn't often predictive of how these kids were going to do long term, that there were environmental factors, family factors, et cetera, that also played a role. So I think now we recognize that we can focus on two streams. As Jerry mentioned, we can look at the babies with the most severe forms of BPD that we think do have a higher risk of long-term complications. But we recognize there's other babies who have very little symptoms in the NICU and end up with really severe asthma and complications at a year or two. And we don't want to miss those kids either. And we also want to look at kids who are dying before 36 weeks post-conceptional age. So the exciting part about it is we're going to identify all the kids who really have problems at 1 to 2 years of age and look at that follow-up data and work our way backwards to see what happened over that first year or two, to see what happened in the NICU, and to see if we can establish a more robust set of risk factors. So I think we're really excited about those prospects, and that will be really integral on what the database has to tell us. And my final question is for Heike. You know, with your blood pressure measurements, you know, I often say to my fellows, I actually don't care what the number is for blood pressure. Just tell me 3 things. Are they urinating? Do they look perfused? Are their little toes pink? And do they have metabolic acidosis on their blood gas? And if the answer to those 3 questions is no, and they seem fine, and I don't really care what the number is. but I'm curious to see if with using the database, we can take your numbers, because I mean, you deserve all the credit in the world. If one more person tells me blood pressure equals mean gestational age, I'm going to die. Especially when we found out it was a nurse from rural London who made that up on her own with no data. So I'm curious to see if we can bring in some of these other physical findings to support your data, saying that this is the normal range, because this would be the normal ranges when they also have normal urine output, for instance.
Yeah, so that's one of the things we want to look at when we look at the real world data, because there will be more of this data and correlations to come together, not only for the low blood pressure, but also for high blood pressure. For neonatal hypertension is such a difficult area, at least for me to treat. And as you know, Yanis luckily is a pediatric nephrologist, so we will be working on both sides of the coin, so to say. And I think it's a unique opportunity to have these laboratory values and the blood pressure values together. So the main thing will be we'll talk with Campbell and make a very nice plan, because the data analysis plan in the beginning, like we did with our systematic review, I think that will be a key issue. And we have got a good, robust plan already, which we can build up on. So we have thought about that already.
I think that's great, especially the hypertension. You know, we always sort of pick, well, if they go over 100, we better treat it. But we actually don't look at the end organ damage and if there's any evidence of that.
And so forth.
Yeah.
And this data will be built in there as well. So I think there's a huge opportunity there.
Excellent.
Hi, Mark Turner. I wonder if so we've got 35 minutes, so maybe I can make some comments as well, and ask a question. So my observation is that each of the groups has taken a long time to refine what they're doing, and that patience really pays off. And one example of that is the pain group. And Rebecca told us about this meeting and this meeting, but I think that is really yielding fruit. And perhaps one bit of cross-fertilization that could take place is that story you've been telling about which bit of reliability and which bit of the validity picture matters. And I wonder whether, say, the seizures group or the pain or the blood pressure group, other groups could learn from that experience of homing in on what really matters to regulators or to investigators, and so that we can then use that experience, because it's hard won. Those discussions with regulators and industry have taken some time, but there is an idea as to what sort of endpoint we should be using. And to John's point, what do we need to look along the way, and how do we check that those observations are going to be useful?
I think that's a really important point. There's so much commonality around the properties of measurements and the processes around the different types of validation. And in fact, as you say, half the work was working out what we were claiming as valid and what type of reliability or interpretability we could use. And clearly this would be highly relevant to the other disciplines, and in fact almost forms one of the core functions of many of the groups here, alongside perhaps the communication, which is very clearly cross-cutting. So perhaps this kind of concept of having these kind of cross-disciplinary groups as one key aspect, kind of intersecting with all of these particular work groups, would really facilitate the learning, because you just wouldn't want to start again if you didn't have to. And so much of it is common. But how to communicate that and how to actually bring that together in the early stages is probably something that needs a bit of thinking about, because actual infrastructure of these are some of the processes you go through to kind of start probably would, enable people to get going quickly. Certainly, we've been communicating together around the data management plans and these different things, and it would be credible start again when actually we could rely on Heike to share much of her learning. So it'd be worth thinking about how we can do that better.
Rebecca, I heard you talk about
using touch of the parent to relieve pain. We have a NICU parent leader named Jamile Jackson. She got Gates funding for it was a project around kangaroo care. Do you have any interest in exploring kangaroo care as another method? Or I understand why you go with touch because it's easier and it's simpler to.
So I think kangaroo care would be really interesting, and it's definitely something we should look at. I think one of the aspects of kangaroo care is people often assume it's a kind of static hold, but it's not. It's a dynamic moving hold. And if you actually look at what parents are doing when they're giving kangaroo care, they're stroking these babies at the speed which will optimally activate a set of fibers, which we think we're activating through the touch. So the reason we chose it is not because it was necessarily easier to implement, although that's a huge advantage to be able to give kind of clear timelines, is we think it might be one of the underpinning mechanisms that drives this. And no doubt kangaroo care would add a whole other level of additional benefits that would be on top. And so, well, if the measures work, then we can reliably start testing the interventions.
I will connect you with Jamile. Thank you.
Other questions? Okay. Well.
So this is it.
Yeah. So it's been awesome, really. No, really, it's been really, really awesome to have you all come in and spend a couple of days with us. Really nice to see you all. As I was saying, I had met anybody of you at all except for John once. So it was it was really, really nice seeing all of our wonderful members. Traveled across. Some of them came from Japan, UK, other countries. I'm not going to take too much of your time except say thank you. It was really nice to see you. And another point would be we are dedicated to fulfilling our ultimate mission of INC, that is meeting unmet drug development needs of neonates. We do look forward to seeing you again next year. Any
Yes,
comments,
and well, this, as you heard in the beginning, this meeting we wanted to have in 2020. We had to wait a little bit, but I think with the data updates, it was totally worth it. So this is a testament again, the hardship that we overcame through the pandemic, but a testament to the commitment that you have, and we reciprocate with the commitment we give you. So next year is going to be even better and bigger, and we're going to start having some really cool answers to share. So stay tuned.
Yep, something really big coming up next year.
Yes.
And yes, stay tuned, please. Thank you. Thank you again. And looking forward to seeing you all again next year.